Hello there, and welcome to the Sleepy Science channel. Tonight, we'll be exploring something extraordinary that exists within you at this very moment. Something invisible to the naked eye, yet responsible for everything you are. We'll be journeying into the world of genetics, the elegant code written inside every cell of your body. A code that connects you to your parents, to your ancestors stretching back millennia, and to every living thing on Earth. Genetics is the story of inheritance, of tiny molecular machines copying themselves with breathtaking precision, of instructions passed down through generations. It's a story written in a language older than any human tongue, in molecules so small that billions could fit on the head of a pin. Yet this microscopic alphabet shapes everything from the color of your eyes to the rhythm of your heartbeat. If you enjoy these quiet explorations, I invite you to like and subscribe, or share a simple thought down in the comments. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is rest, settle in, and allow the calm wonder of the genetic world to carry you gently towards sleep. Let's begin. Every cell contains your complete genetic blueprint. Within your body right now are trillions of cells. Each one, whether it's a skin cell on your fingertip or a neuron firing in your brain, contains an identical copy of your genetic information. This complete instruction manual sits coiled in the nucleus, the command center of each cell, waiting to be read. The same blueprint exists in every cell. Yet somehow, a heart cell knows to beat, and a stomach cell knows to digest. The instructions are universal, but each cell reads only the chapters relevant to its purpose. This genetic blueprint is remarkably compact. If you could unwind all the DNA from a single cell and stretch it out, it would measure about 2 meters long. Yet it fits comfortably inside a nucleus, just a few micrometers across. The packaging is exquisite, a feat of molecular origami that allows immense information to occupy impossibly small spaces. Scientists estimate that your body contains enough DNA that if it were all connected end to end, it would stretch from Earth to the Sun and back multiple times. All of this carries the same message, the same instructions, faithfully copied billions upon billions of times. Every cell is a library, containing your entire genetic encyclopedia. DNA resembles a twisted ladder, reaching into microscopic depths. The molecule that carries genetic information has a structure both elegant and functional. DNA, deoxyribonucleic acid, forms a double helix, a shape like a twisted ladder spiraling upward. The sides of this ladder are made of sugar and phosphate molecules, providing a stable backbone. The rungs between them are pairs of smaller molecules called bases, fitting together with remarkable specificity. This twisted ladder was first visualized in the 1950 seconds, when scientists finally understood its architecture. The double helix immediately suggested how genetic information could be copied. Each side of the ladder serves as a template for creating a new strand, allowing the molecule to replicate itself with precision. The elegance of this design lies in its simplicity, a structure that is both stable enough to preserve information and flexible enough to be read and copied. Under a powerful microscope, DNA appears as fine threads, chromatin, tangled within the nucleus. But zoom in further, past the limits of light microscopy, and the double helix emerges. 2 strands intertwined, winding around each other in a gentle spiral, a molecular staircase spiraling down into the heart of heredity. 4 chemical letters spell out all genetic instructions. The information in DNA is written in an alphabet of just 4 letters. These are the nucleotide bases: adenine, thymine, guanine, and cytosine, abbreviated as A, T, G, and C. Every instruction, every gene, every trait that makes you uniquely you is spelled out using only these 4 chemical letters. It's an alphabet simpler than any human language, yet capable of encoding extraordinary complexity. These bases pair with absolute specificity. Adenine always pairs with thymine. Guanine always pairs with cytosine. This pairing rule, discovered through careful chemical analysis, is the key to DNA's ability to copy itself. When the double helix unzips down the middle, each strand serves as a template. An A on one strand will attract a T to the new strand being built. A G will attract a C. The result is 2 identical copies where there was once one. The sequence of these 4 letters determines everything. A gene might begin with ATGCG ATCG and continue for thousands of letters, each position critical. Change a single letter, and you might change an entire trait, or you might change nothing at all. Some sequences are instructions for building proteins. Others are control switches, determining when and where genes are turned on or off. Still others seem to serve structural purposes, or functions we're only beginning to understand. Genes are recipes written in this 4-letter code. Within the long strands of DNA are special sections called genes. Each gene is a recipe, a set of instructions for building a specific protein. Proteins are the workhorses of the cell, carrying out nearly every function necessary for life. Some proteins form structures, like the keratin in your hair or the collagen in your skin. Others are enzymes that speed up chemical reactions. Still others are hormones, signaling molecules, or molecular machines. A gene might be a few hundred base pairs long, or it might stretch across thousands. When a cell needs a particular protein, molecular machinery reads that gene's sequence. The 4-letter code is translated into a chain of amino acids, the building blocks of proteins. Each 3-letter sequence in the DNA codes for one amino acid. ATG might code for methionine. GGC might code for glycine. The sequence determines the order of amino acids, and that order determines how the protein folds and what it does. You have approximately 20,000 genes in your genome. Some are active in every cell, housekeeping genes that maintain basic functions. Others are specialists, turned on only in certain cells or at certain times. The gene for insulin is read primarily in pancreatic cells. The gene for hemoglobin is active in developing red blood cells. Each gene waits in the DNA, ready to be transcribed when its protein is needed. Chromosomes package DNA into organized bundles. Your genome, all 3 billion base pairs of it, is organized into structures called chromosomes. These are long strands of DNA wrapped around proteins called histones, forming tight bundles that help organize and protect the genetic material. Humans have 46 chromosomes in total, 23 pairs. 1 chromosome from each pair came from your mother, the other from your father. Under a microscope, chromosomes are most visible when a cell is dividing. They condense into thick, X-shaped structures, each one a tightly coiled package of DNA. When the cell isn't dividing, chromosomes become looser, spreading out into a tangle of chromatin that fills the nucleus. This loose form allows the cell's machinery to access the DNA, reading genes and copying information as needed. Each chromosome contains hundreds or thousands of genes, along with vast stretches of DNA that don't code for proteins. Some of these non-coding regions act as regulatory elements, controlling when and how genes are expressed. Others are structural, helping chromosomes maintain their shape and separate properly during cell division. The largest human chromosome, chromosome 1, contains more than 240 million base pairs and thousands of genes. The smallest, chromosome 21, is still home to hundreds of genes despite being much shorter. You inherited half your DNA from each parent. You are a genetic mosaic, a combination of your mother and your father. When you were conceived, you received 23 chromosomes from your mother's egg and 23 from your father's sperm. These paired up to form the 46 chromosomes in every cell of your body. This means roughly half your DNA came from each parent, blending their genetic information to create someone entirely new. But inheritance isn't simply a matter of mixing. The chromosomes you received from your parents were themselves shuffled versions of the chromosomes they received from their parents. Before sperm and egg cells form, chromosomes pair up and exchange segments in a process called recombination. This shuffling means that the chromosome you inherited from your mother isn't identical to either of the chromosomes she inherited from your grandparents. It's a patchwork, pieces from both woven together. This process ensures that siblings aren't identical, even though they share the same parents. You and your sibling each received different combinations of chromosomes, different shuffled segments. The only exception is identical twins, who develop from a single fertilized egg that splits in two. They share the same genetic blueprint, the same chromosomes, the same sequences. But even identical twins accumulate small differences over time as their cells divide and their genomes experience different mutations. Identical twins share identical genetic sequences. Identical twins begin as one. A single sperm fertilizes a single egg, creating a single embryo with a unique genetic code. Then, in the earliest days of development, that embryo splits into two. Each half continues developing independently, but both carry the exact same DNA sequence. They are genetic duplicates, a natural experiment in the interplay between genes and environment. Despite sharing identical genomes, identical twins are not truly identical in every way. As they grow, small differences emerge. Their fingerprints, formed by random patterns during development, are similar, but not the same. Their experiences shape them differently. One might develop a scar the other doesn't have. One might be exposed to different environmental factors. Over time, even their DNA begins to diverge slightly, as cells accumulate random mutations. Yet the similarities remain striking. Identical twins often look remarkably alike. They're more likely to share personality traits, talents, and even preferences than fraternal twins or ordinary siblings. Researchers study identical twins to understand which traits are influenced more by genetics and which by environment. When twins raised apart still share interests or abilities, it suggests genetics play a strong role. When they differ, it highlights the power of experience and choice. Fraternal twins are genetically like any siblings. Fraternal twins, unlike identical twins, develop from two separate eggs, fertilized by two separate sperm. They share a womb and a birthday, but genetically, they're no more similar than any other siblings. Each received a different combination of chromosomes from their parents, a different shuffling of the genetic deck. Fraternal twins can be the same sex or different sexes. They might look very similar, or they might look quite different from each other. Because they share approximately 50% of their variable DNA, just like any siblings, they can inherit different traits. One might have their mother's eye color, while the other has their father's. One might be tall, while the other is shorter. The frequency of fraternal twins varies across populations and families. Some families seem to have twins in multiple generations, suggesting a genetic component to the likelihood of releasing multiple eggs during ovulation. Identical twins, by contrast, occur randomly at roughly the same rate across all populations. The splitting of an embryo appears to be a chance event, not influenced by genetics. Genetic variation makes every human unique. Though all humans share the vast majority of their DNA, no two people are exactly alike, except identical twins. The human genome contains billions of base pairs, and at millions of these positions, the sequence can vary. One person might have an A, where another has a G. These small differences, called single nucleotide polymorphisms, or SNPs, are scattered throughout the genome. Most of these variations occur in regions between genes or within genes, in ways that don't change the protein produced. They're silent differences, contributing to uniqueness without necessarily affecting function. But some variations do matter. A single base pair change might alter an enzyme's efficiency, affect how a receptor responds to a signal, or change the structure of a protein. These variations accumulate over generations, spreading through populations. Some become common, while others remain rare. The pattern of variation across a genome can reveal ancestry, as different populations have different frequencies of particular variants. It can also reveal relationships, as close relatives share more genetic variants than distant ones. Your unique combination of variants, inherited from your parents and their parents before them, is what makes you genetically distinct. Mutations are changes in the genetic code. Mutations are alterations to the DNA sequence. They can occur when DNA is copied and a mistake slips through the cell's proofreading mechanisms. They can be caused by environmental factors like radiation or certain chemicals, or they can happen randomly, a consequence of the inherent chemistry of DNA. Most mutations are harmless. They occur in non-coding regions or change a DNA letter in a way that doesn't alter the protein produced. Some mutations are even beneficial, providing an advantage that helps an organism survive and reproduce. Over many generations, beneficial mutations can spread through a population, driving evolution. But some mutations are harmful, disrupting important genes or regulatory regions. A mutation might cause a protein to fold incorrectly, rendering it non-functional. Another might interfere with a gene's control switches, causing it to be expressed at the wrong time or in the wrong amount. Harmful mutations can lead to genetic diseases if they occur in critical genes. Every person carries many mutations that arose in their parents' sperm or egg cells, or in the early divisions of the embryo. These are new mutations, not inherited from previous generations. Most will be harmless or silent. A few might contribute to traits or health conditions, and some will be passed on to the next generation, becoming part of the ongoing story of genetic variation. Some mutations cause genetic disorders. When a mutation disrupts a critical gene, it can lead to a genetic disorder. Thousands of such conditions are known, each caused by changes to specific genes. Some genetic disorders are caused by a single mutation in a single gene. Sickle cell anemia, for instance, results from a single base pair change in the gene for hemoglobin. This tiny alteration causes red blood cells to deform into a sickle shape, leading to pain and complications. Other genetic disorders result from missing or extra chromosomes. Down syndrome occurs when a person has 3 copies of chromosome 21 instead of 2. The extra genetic material disrupts normal development, leading to characteristic features and intellectual disability. Turner syndrome, by contrast, occurs when one of the X chromosomes is missing or partially deleted in females. Many genetic disorders are recessive, meaning a person must inherit 2 copies of the mutated gene, one from each parent, to develop the condition. Carriers, who have 1 normal copy and 1 mutated copy, are typically healthy. But if 2 carriers have children, there's a chance their child will inherit both mutated copies. This is how conditions like cystic fibrosis or Tay-Sachs disease are passed down through families. Some traits follow simple inheritance patterns. Gregor Mendel, a 19th century monk, discovered the basic principles of inheritance by breeding pea plants. He noticed that certain traits, like flower color or seed shape, followed predictable patterns. When he crossed a plant with purple flowers with one with white flowers, all the offspring had purple flowers. But when he crossed those offspring with each other, white flowers reappeared in about 1/4 of the next generation. Mendel concluded that traits are controlled by discrete units of inheritance, which we now call genes. Each parent contributes one copy of each gene. Some versions, or alleles, are dominant, masking the effect of other alleles. Other alleles are recessive, only visible when two copies are present. This simple model explains many human traits. Widow's peak, a V-shaped hairline, is often dominant over a straight hairline. The ability to roll your tongue is dominant over the inability. But most human traits are far more complex, influenced by multiple genes and environmental factors. Eye color, height, intelligence, and personality don't follow Mendel's simple patterns. They arise from the interplay of many genes, each contributing a small effect, along with nutrition, experience, and chance. Eye color involves multiple genes working together. Eye color was once thought to be a simple trait, determined by a single gene with brown dominant over blue. Parents with brown eyes, the thinking went, could have a blue-eyed child if both carried a recessive blue allele. But the reality is more nuanced. Eye color is influenced by multiple genes, each contributing to the amount and distribution of melanin in the iris. The OCA2 and HERC2 genes on chromosome 15 play major roles. Variations in these genes can reduce melanin production, leading to blue or green eyes instead of brown. But other genes also contribute, fine-tuning the shade and adding complexity. This is why two brown-eyed parents can sometimes have a blue-eyed child, but also why two blue-eyed parents very rarely have a brown-eyed child. The spectrum of eye colors, from deep brown to pale blue, from green to hazel, reflects this genetic complexity. Environmental factors even play a role. The amount of light exposure can slightly influence eye color in infancy. Babies are often born with blue or gray eyes that darken over the first year as melanin production ramps up. The final eye color, settled usually by age 3, is a product of multiple genes expressing themselves during development. Height is influenced by hundreds of genetic variants. Height is one of the most heritable human traits. Tall parents tend to have tall children, and short parents tend to have short children. Yet height doesn't follow a simple inheritance pattern like Mendel's pea plants. Instead, it's a polygenic trait, influenced by hundreds or even thousands of genetic variants scattered across the genome. Each variant has a tiny effect, nudging height up or down by millimeters. Some affect growth hormones. Others influence the development of bones or cartilage. Still others play roles in nutrition and metabolism, indirectly affecting how much a person grows. When you add up the effects of all these variants, plus environmental factors like childhood nutrition and health, you get the wide range of human heights. Researchers have identified hundreds of specific genetic variants associated with height. Together, these variants explain about 80% of the variation in height among individuals. The remaining 20% comes from environment and chance. Two siblings with similar genetics might still differ in height if one had better nutrition or fewer illnesses during childhood. Height is a reminder that genes provide a blueprint, but the final outcome depends on how that blueprint is built in the real world. Genes influence, but don't determine complex traits. Many human characteristics, from intelligence to personality to susceptibility to diseases, have genetic components. Studies of twins and families show that genes influence these traits. Identical twins raised apart are often more similar in IQ or temperament than fraternal twins raised together, suggesting genetics play a role. But genes are not destiny. Environmental factors matter enormously. A child with genetic potential for high intelligence might not reach that potential without proper nutrition, education, and stimulation. Someone with genetic risk factors for heart disease might avoid illness through healthy lifestyle choices. Genes load the gun, as the saying goes, but environment pulls the trigger. This interplay between nature and nurture is central to understanding genetics. Genes provide tendencies, predispositions, and possibilities. They influence how your body responds to the environment, how easily you learn certain skills, how likely you are to develop certain conditions, but they don't write your story in stone. Your choices, your experiences, and chance events all shape who you become. Gene expression determines when and where genes are active. Having a gene is not the same as using it. Every cell in your body contains the same genes, yet a neuron is very different from a muscle cell. This difference arises from gene expression, the process by which genes are turned on or off. A neuron expresses genes for neurotransmitters and ion channels. A muscle cell expresses genes for contractile proteins. Each cell type has its own expression profile, reading different chapters of the shared genetic manual. Gene expression is controlled by regulatory regions in the DNA and by proteins that bind to these regions. Transcription factors, proteins that recognize specific DNA sequences, can activate or repress genes. Enhancers and silencers, stretches of DNA sometimes located far from the genes they control, fine-tune expression levels. The cell can respond to signals from outside, turning genes on or off in response to hormones, nutrients, or stress. This regulation is exquisitely precise. During development, waves of gene expression sweep through the embryo, turning genes on and off in carefully choreographed sequences. These patterns determine where a head forms, where limbs grow, which cells become heart, and which become brain. Errors in gene regulation can lead to developmental abnormalities or diseases. But when everything works correctly, the result is the astonishing complexity of a functioning organism, all from the same genetic starting point. Epigenetics adds another layer of genetic control. Beyond the DNA sequence itself, there's another level of information, epigenetics. Literally, above genetics, refers to chemical modifications to DNA and the proteins it wraps around. These modifications don't change the sequence of base pairs, but they do affect whether genes are expressed or silenced. One common epigenetic mark is DNA methylation, the addition of a small chemical group to certain bases. When a gene's promoter region is heavily methylated, the gene is typically turned off. The cell's machinery can't access the DNA to read the gene. Conversely, removing methylation can activate a silent gene. Histone modifications, changes to the proteins that DNA coils around, can also open up or close off regions of the genome. Epigenetic marks can be influenced by environment. Diet, stress, toxins, and experiences can all alter epigenetic patterns. Some of these changes can even be passed to the next generation, though this remains an area of active research. The idea that your experiences might somehow mark your DNA and affect your children or grandchildren is both fascinating and controversial. What's clear is that epigenetics provides a flexible layer of control, allowing cells to respond to their environment without changing their underlying genetic code. It's a way for nature and nurture to interact at the molecular level. RNA serves as the messenger between DNA and proteins. DNA stores genetic information, but it doesn't build proteins directly. That job belongs to RNA, ribonucleic acid, a close chemical cousin of DNA. When a gene needs to be expressed, an enzyme called RNA polymerase reads the DNA sequence and creates a complementary RNA copy. This messenger RNA, or mRNA, carries the gene's instructions from the nucleus out to the cytoplasm. In the cytoplasm, molecular machines, called ribosomes, read the mRNA sequence. They translate the genetic code into a chain of amino acids, assembling the protein one building block at a time. The process is remarkably fast and accurate. A single ribosome can add several amino acids per second. Multiple ribosomes can read the same mRNA simultaneously, producing many copies of a protein from a single message. RNA is more versatile than DNA. Some RNA molecules don't code for proteins at all, but instead play regulatory roles. MicroRNAs bind to mRNA molecules and prevent them from being translated, fine-tuning protein production. Ribosomal RNA forms the core of ribosomes themselves. Transfer RNA molecules ferry amino acids to the ribosome during protein synthesis. This diverse family of RNA molecules shows that genetic information flows through multiple channels, not just from DNA to protein. Proteins perform nearly every function in your cells. If DNA is the blueprint, proteins are the builders, the workers, the machinery of life. They come in countless varieties, each with a specific shape and function. Some proteins are enzymes, speeding up chemical reactions that would otherwise take years to complete. Digestive enzymes break down food. Metabolic enzymes extract energy. DNA polymerase copies genetic information. Other proteins provide structure. Collagen forms tough fibers in skin, tendons, and bones. Keratin creates hair and nails. Actin and myosin filaments slide past each other to create muscle contractions. Structural proteins give cells their shapes and hold tissues together. Still other proteins are messengers and receptors. Insulin signals cells to take up glucose. Antibodies recognize foreign invaders. Receptor proteins on cell surfaces detect hormones and neurotransmitters, triggering responses inside the cell. Proteins transport molecules, transmit signals, regulate genes, defend against disease, and perform thousands of other tasks. Life, at its most fundamental level, is protein chemistry orchestrated by genetic instructions. Each protein folds into a specific three-dimensional shape. A protein begins as a linear chain of amino acids, but it doesn't stay that way. Almost immediately, it begins folding into a complex three-dimensional shape. This shape is critical to function. An enzyme's active site must fit its substrate like a lock and key. An antibody's binding region must match its target precisely. If a protein misfolds, it may not work, or worse, it may become harmful. The sequence of amino acids determines how the protein folds. Some amino acids are hydrophobic, avoiding water and clustering together in the protein's interior. Others are hydrophilic, staying on the surface where they can interact with the cell's watery environment. Chemical bonds and interactions between amino acids stabilize the folded structure. Cells have specialized proteins called chaperones that help other proteins fold correctly. They prevent tangling and misfolding, guiding new proteins into their proper shapes. When proteins do misfold, other systems recognize and destroy them. Failure of these quality control systems can lead to disease. In Alzheimer's disease, misfolded proteins accumulate in the brain. In cystic fibrosis, a misfolded protein fails to reach the cell's surface where it's needed. Protein folding is one of biology's great puzzles. Predicting how a sequence will fold remains difficult. though recent advances in artificial intelligence have made progress. Understanding folding could revolutionize medicine, allowing scientists to design proteins with specific functions or to correct misfolding diseases. The genetic code is nearly universal across all life. One of the most profound discoveries in genetics is that the code is universal. The same triplet of DNA letters that codes for a specific amino acid in a human also codes for that amino acid in a bacterium, a mushroom, or a whale. ATG codes for methionine. TGG codes for tryptophan. With a few rare exceptions, all living things use the same genetic dictionary. This universality is powerful evidence of common ancestry. All life on Earth, from the simplest microbe to the most complex animal, inherited this code from a shared ancestor billions of years ago. The code has been preserved through eons of evolution, because changing it would be catastrophic. Nearly every protein depends on the code being read correctly. A change would scramble every gene at once. The universality also makes genetic engineering possible. Scientists can take a human gene and insert it into bacteria, which will read the gene and produce the human protein. This is how insulin is manufactured today. Bacteria, carrying the human insulin gene, churn out the hormone in fermentation vats, providing a supply for diabetics. The fact that bacteria can read human genes is a testament to our shared genetic heritage. You share genes with every living organism. You have more in common with a fruit fly than you might think. Humans and flies share thousands of genes that perform similar functions. Many genes involved in basic cell processes, metabolism, and development are conserved across vast evolutionary distances. Even plants share genes with animals. We all need to convert energy, build proteins, and repair damage. The degree of similarity reflects evolutionary relationships. You share about 99% of your DNA sequence with chimpanzees, your closest living relatives. You share roughly 90% with mice, 85% with dogs, and about 60% with fruit flies. Even baker's yeast shares about 30% of its genes with humans. These shared genes tend to be ancient and essential, coding for fundamental processes necessary for life. This genetic continuity reveals that life is a tapestry woven from common threads. Differences arise from changes to existing genes and from the evolution of new genes, but the foundation remains the same. When scientists study genes in yeast or worms or flies, they're often learning about processes relevant to human health. Many human disease genes were first identified by studying their counterparts in simpler organisms. Evolution shapes genomes over millions of years. Genomes are not static. They change over time through mutation, natural selection, and genetic drift. A beneficial mutation might help an organism survive and reproduce, passing the mutation to more offspring. Over generations, that mutation becomes more common. Harmful mutations tend to disappear as individuals carrying them have fewer offspring. Natural selection acts on variation. In a population with genetic diversity, some individuals will be better suited to their environment. They'll be more likely to survive predators, resist disease, find food, and attract mates. Their genes will spread through the population. Over many generations, this process shapes the genome, favoring variants that enhance survival and reproduction. Evolution also involves neutral changes. Some mutations neither help nor harm. They spread through populations randomly, by genetic drift. Comparing genomes across species reveals both the footprints of selection and the noise of drift. Regions of the genome under strong selective pressure change slowly, because mutations there are usually harmful. Regions with less functional constraint change faster, accumulating neutral mutations over time. The human genome carries the history of our evolution. Comparisons with chimpanzee and other primate genomes reveal what changed as our lineage diverged. Changes in genes affecting brain size, speech, and cognition appear particularly important. Our genome is a palimpsest, layers of evolutionary history, written, overwritten, and preserved in the DNA. Humans share more than 99% of their DNA. All humans are remarkably similar genetically. Any two people, chosen at random from anywhere on Earth, will have DNA sequences that are more than 99% identical. The differences that make us unique, that create the diversity of human appearance and ability, come from less than 1% of the genome. This similarity reflects our recent common ancestry. Modern humans emerged in Africa roughly 300,000 years ago. Compared to many species, we're young and haven't had much time to accumulate differences. Genetic studies suggest that all living humans descend from a population that may have numbered just a few thousand individuals at certain points in the past. This bottleneck reduced our genetic diversity. The small differences that do exist are concentrated in certain regions of the genome. Some of these differences affect visible traits like skin color, hair texture, and facial features. Others influence disease, susceptibility, or drug metabolism, but the vast majority of our genome is identical across all human populations. This genetic unity underscores a fundamental truth. The concept of distinct human races has no biological basis. We are one species with minor variations. Ancient DNA reveals our evolutionary past. DNA doesn't last forever. In most conditions, it degrades within thousands of years, but in cold, dry, or otherwise favorable environments, fragments can persist much longer. Scientists have extracted and sequenced DNA from ancient bones and teeth, opening a window into the past. Neanderthal DNA, recovered from fossils tens of thousands of years old, revealed that these extinct humans interbred with modern humans. Most people today, of European and Asian descent, carry 1 to 2% Neanderthal DNA. Denisovans, another extinct human group known mostly from fragmentary remains in Siberia, also contributed DNA to modern human populations, particularly in Southeast Asia and Oceania. Ancient DNA from humans who lived thousands of years ago shows how populations migrated and mixed. It reveals the spread of agriculture, the movement of peoples, and the genetic impact of historical events. DNA from Egyptian mummies, Viking burials, and prehistoric hunter-gatherers tells stories written in genes. This field, paleogenomics, combines genetics with archaeology and anthropology. It's rewriting human prehistory, revealing complexity that artifacts alone could never show. Every ancient genome is a voice from the past, carrying genetic echoes of lives lived long ago. Mitochondria have their own separate DNA. Inside each of your cells, alongside the nucleus, are hundreds of tiny organelles called mitochondria. These are the power plants of the cell, converting nutrients into energy. Remarkably, mitochondria have their own DNA, separate from the DNA in the nucleus. This mitochondrial DNA, or mtDNA, is much smaller than nuclear DNA, just 37 genes compared to about 20,000. But it's essential. The presence of mitochondrial DNA is a relic of evolution. Mitochondria were once free-living bacteria that were engulfed by an ancient ancestor of all complex cells. Instead of being digested, they formed a symbiotic relationship, providing energy in exchange for a protected environment. Over billions of years, most of their genes transferred to the nucleus, but a small genome remained. Mitochondrial DNA is inherited differently than nuclear DNA. You receive all your mitochondria from your mother, passed down through the egg cell. Sperm contribute essentially no mitochondria to the embryo. This means your mtDNA is a direct maternal lineage, identical to your mother's and your mother's mother's, stretching back through generations. Researchers use mtDNA to trace maternal ancestry and to study human migration patterns over millennia. Mitochondrial DNA traces maternal ancestry. Because mtDNA passes from mother to child without recombination, it's a powerful tool for tracing lineages. Mutations in mtDNA accumulate slowly over generations, creating distinct lineages. By comparing mtDNA sequences from people around the world, geneticists have constructed a maternal family tree for all of humanity. This tree traces back to a woman nicknamed Mitochondrial Eve, who lived in Africa roughly 150,000 to 200,000 years ago. She's not the only woman who lived then, but she's the one whose mitochondrial lineage survived to the present. Every person alive today carries mtDNA descended from hers. Over time, her descendants dispersed across Africa and eventually around the world, accumulating mutations that now mark different maternal lineages. These lineages, called haplogroups, reveal migration patterns. Certain haplogroups are common in specific regions, reflecting the movements of ancient populations. By tracing your maternal haplogroup, you can glimpse the path your maternal ancestors took from Africa to other continents, through ice ages and changing climates, until they arrived at wherever your family is today. Y chromosomes trace paternal ancestry. Just as mtDNA traces maternal lines, the Y chromosome traces paternal lines. Only males have a Y chromosome, inherited from father to son. Like mtDNA, most of the Y chromosome doesn't recombine, preserving lineages across generations. Mutations accumulate, creating distinct Y chromosome haplogroups. Genetic studies of the Y chromosome have identified a Y chromosomal Adam, the most recent common patrilineal ancestor of all living men. He lived in Africa between 200,000 and 300,000 years ago. Like mitochondrial Eve, he wasn't the only man alive, but his Y chromosome lineage is the one that survived. Y chromosome haplogroups map the dispersal of human populations from a male perspective. They reveal ancient migrations, the spread of languages and cultures, and even historical events. The Y chromosome can show how Vikings spread across Europe, how Genghis Khan's lineage became widespread in Asia, and how different populations mixed over time. Together, mitochondrial DNA and Y chromosome studies provide complementary views of human history. They show that modern humans originated in Africa and spread across the globe, diversifying into the populations we see today, while remaining a single, interconnected species. Some genes are dominant, while others are recessive. When you inherit two different versions of a gene, one from each parent, which one determines your trait? Sometimes one allele is dominant, meaning its effect is visible even if you have only one copy. The other allele is recessive, its effect masked unless you have two copies. A classic example is the gene for Huntington's disease. This devastating neurological disorder is caused by a dominant mutation. If you inherit one mutated copy, you will eventually develop the disease, even if your other copy is normal. Dominant disorders are often rare because they usually reduce reproduction, preventing the allele from spreading. Recessive disorders, by contrast, can be passed through families for generations without causing disease. Carriers with one normal and one mutated copy are healthy. But if two carriers have children, each child has a 25% chance of inheriting both mutated copies and developing the disorder. Cystic fibrosis, sickle cell anemia, and Tay-Sachs disease follow this pattern. Understanding dominance and recessiveness is key to genetic counseling. Couples can be tested to see if they carry mutations for recessive disorders. If both are carriers, they can make informed decisions about family planning, prenatal testing, or reproductive technologies. Some people carry protective gene variants. Not all mutations are harmful. Some provide advantages, protecting against disease or enhancing survival. People with a specific mutation in the CCR5 gene are resistant to HIV infection. The mutation prevents the virus from entering immune cells, providing near-complete protection. It's relatively common in European populations, possibly because it also provided some protection against historical plague epidemics. Another example is the sickle cell trait. Carrying one copy of the sickle cell mutation doesn't cause disease, but does provide protection against malaria. In regions where malaria is common, this protection is a significant advantage, explaining why the sickle cell allele remains frequent despite causing disease in those who inherit two copies. Some gene variants affect lactose tolerance. Most adult mammals lose the ability to digest lactose, the sugar in milk, after weaning. But in human populations with a long history of dairy farming, mutations arose that keep the lactase enzyme active throughout life. These mutations became common because they allowed adults to consume milk, a valuable source of nutrition. Today, lactose tolerance is widespread in European, some African, and Middle Eastern populations, but rare elsewhere. These examples show that genetic variation isn't just about disease risk. It's also about adaptation, resilience, and the diverse ways humans have responded to their environments over millennia. Blood type is determined by genes on chromosome 9. Your blood type, A, B, AB, or O, is determined by a gene on chromosome 9. This gene codes for an enzyme that adds specific sugars to proteins on the surface of red blood cells. The A allele adds one type of sugar, creating type A blood. The B allele adds a different sugar, creating type B. The O allele produces a non-functional enzyme, adding no sugar, resulting in type O. Because you inherit one allele from each parent, you can have combinations. AA or AO gives you type A blood. BB or BO gives type B. AB gives type AB, and OO gives type O. A and B are codominant, meaning both are expressed if you have one of each. O is recessive. Blood type matters for transfusions. If you receive blood with markers your body doesn't recognize, your immune system attacks it. Type O blood lacks both A and B markers, so it can be given to anyone in emergencies. Type AB blood has both markers, so people with AB blood can receive any type. These compatibility rules have saved countless lives since blood transfusions became routine medical practice. Blood type also reveals inheritance patterns. If both parents have type O blood, all their children must have type O. If one parent has type AB, their children cannot have type O. These patterns can confirm parentage or rule it out, though DNA testing is now more definitive. The Rh factor adds another layer to blood type. In addition to A, B, AB, or O, blood types are also classified as positive or negative based on the Rh factor, another protein on red blood cells. If you have the Rh factor, you're Rh positive. If not, you're Rh negative. About 85% of people are Rh positive. Rh status usually doesn't matter, except during pregnancy. If an Rh negative mother carries an R positive baby, her immune system might recognize the baby's red blood cells as foreign and produce antibodies against them. In a first pregnancy, this usually doesn't cause problems, but in subsequent pregnancies with Rh positive babies, those antibodies can attack the baby's blood cells, causing serious complications. Modern medicine prevents this with Rh immunoglobulin, an injection given to Rh-negative mothers during and after pregnancy. This treatment prevents the mother's immune system from producing antibodies, protecting future babies. It's a triumph of understanding genetics and immunology. The Rh factor is inherited through a dominant recessive pattern. Rh positive is dominant, so you need only one copy of the Rh gene to be positive. Ra negative people have 2 copies of the non-functional version. This is why 2 Rh negative parents can only have Rh negative children, but 2 Rh positive parents might have Rh negative children if both are carriers of the recessive allele. Genetic testing can reveal disease risks. Advances in DNA sequencing have made genetic testing widely available. For a few $100s, companies will analyze your DNA and provide information about ancestry, traits, and health risks. Medical genetic testing can identify mutations linked to diseases like breast cancer, colon cancer, or heart disease. Knowing your genetic risks can be empowering. If you carry mutations in the BRCA1 or BRCA2 genes, your risk of breast and ovarian cancer is significantly elevated. Armed with this knowledge, you can increase screening, consider preventive surgeries, or make lifestyle changes to reduce other risk factors. Genetic testing has saved lives by identifying high-risk individuals before cancer develops. But genetic information is also complex and sometimes uncertain. Many genetic risk factors are probabilistic, not deterministic. Having a variant that increases your risk of a disease doesn't mean you'll definitely get it, and lacking that variant doesn't guarantee you won't. Environment, lifestyle, and chance all play roles. There are also ethical considerations. Genetic information is deeply personal. Should insurers or employers have access to it? Could genetic discrimination become a problem? Laws in many countries now protect genetic privacy, but concerns remain. And what about incidental findings? When a test looking for one thing discovers something unexpected and possibly disturbing. These are questions society is still grappling with. Gene therapy aims to correct genetic defects. If a disease is caused by a faulty gene, could you fix it? Gene therapy attempts to do just that, introducing a functional copy of a gene into a patient's cells to replace or supplement a defective one. The idea is elegant, but the execution has been challenging. Early gene therapy trials faced setbacks. Some patients developed immune reactions to the viruses used to deliver genes. A few developed cancer when the new gene inserted into the wrong place in the genome. These complications slowed progress and raised safety concerns. But recent years have seen remarkable successes. Treatments for certain inherited blindnesses have restored vision by delivering a working copy of a gene to retinal cells. Therapies for severe combined immunodeficiency, bubble boy disease, have cured children by correcting the gene in their blood stem cells. Treatments for sickle cell disease and beta thalassemia are showing promising results. Gene therapy is still expensive and available for only a few conditions, but the field is advancing rapidly. New techniques, like CRISPR-based editing, offer precision that earlier methods lacked. The dream of curing genetic diseases by fixing the underlying genetic problem is becoming reality, one condition at a time. CRISPR allows precise editing of DNA. CRISPR, a revolutionary gene editing technology, gives scientists the ability to cut and modify DNA with remarkable precision. It's based on a system bacteria use to defend against viruses. Researchers adapted this natural mechanism into a powerful tool for editing genomes. CRISPR works like molecular scissors guided by a GPS. A short RNA sequence directs the CRISPR protein, Cas9, to a specific location in the genome. There, Cas9 cuts the DNA. The cell's repair machinery then fixes the break, and scientists can influence how it's repaired, deleting unwanted sequences, correcting mutations, or inserting new genes. The technology is faster, cheaper, and more precise than older gene editing methods. It's been used to study genes in organisms from bacteria to humans, to create disease models, and to develop new therapies. Researchers are exploring CRISPR treatments for sickle cell disease, muscular dystrophy, certain cancers, and more. CRISPR also raises ethical questions. If we can edit human embryos, should we? What about editing genes to enhance traits rather than cure disease? The power to rewrite the human genome is both thrilling and daunting, promising medical breakthroughs, but also demanding careful ethical oversight. Cloning creates genetically identical organisms. Cloning, the creation of a genetic duplicate, sounds like science fiction, but is scientific reality. The most famous clone is Dolly the sheep, born in 1996. Scientists took a cell from an adult sheep's udder, extracted its nucleus containing the DNA, and inserted it into an egg cell whose nucleus had been removed. The egg, now carrying adult DNA, was stimulated to divide and develop into an embryo, which was implanted into a surrogate mother. Dolly was born months later, genetically identical to the sheep that donated the cell. Dolly proved that adult cells, despite having specialized into specific types, still carry the complete genome and can be reprogrammed to create an entire organism. Since then, scientists have cloned cattle, pigs, cats, dogs, and other animals. Cloning is used to preserve genetics of valuable livestock, to study diseases, and to explore basic biology. Human cloning remains controversial and is banned in many places. The technical challenges are significant, and the ethical concerns are profound. But the technique reveals something fundamental about genetics. Every cell carries the complete blueprint. Given the right conditions, that blueprint can be reset to the beginning, creating life anew. Identical twins are nature's clones. While artificial cloning is recent, nature has been creating clones for as long as complex life has existed. Identical twins are natural clones, genetically identical individuals arising from a single fertilized egg. After a few cell divisions, the early embryo splits into two separate cell masses. Each continues developing independently, but both carry the same genome. The split that creates twins is a random event, not inherited or controlled. It happens in about 3 or 4 per 1,000 births. Identical triplets or quadruplets are even rarer, occurring when an embryo splits multiple times or when a split embryo splits again. Despite their genetic identity, identical twins are individuals. They have distinct personalities, thoughts, and experiences. Their fingerprints differ. Over time, environmental factors cause their genomes to diverge slightly through epigenetic changes and random mutations. But their fundamental genetic similarity makes them invaluable for research, helping scientists tease apart the contributions of genes and environment to human traits. Stem cells can become many different cell types. Early in development, cells are unspecialized. They haven't yet committed to becoming skin, nerve, muscle, or any specific type. These stem cells have the remarkable ability to differentiate into various cell types. Embryonic stem cells, taken from early embryos, can become any cell type in the body. They're pluripotent, possessing unlimited developmental potential. Adult stem cells, found in various tissues, are more limited. Blood stem cells in bone marrow produce all types of blood cells, but can't become neurons. Skin stem cells replenish skin, but can't form liver. Still, Adult stem cells are crucial for maintaining and repairing tissues throughout life. Scientists have learned to reprogram adult cells back into a stem-like state, creating induced pluripotent stem cells, or iPSCs. By adding specific genes, a skin cell can be transformed into a cell that behaves like an embryonic stem cell, capable of becoming many cell types. This breakthrough avoids the ethical concerns of using embryos and allows creation of patient-specific stem cells for research and potential therapies. Stem cell research holds immense promise. These cells could repair damaged hearts, regenerate spinal cord injuries, treat Parkinson's disease, or grow replacement organs. The genetic programs that allow stem cells to differentiate are being decoded, revealing how genes orchestrate the complex dance of development. Development follows genetic programs. From a single fertilized egg emerges a complete organism with trillions of cells organized into organs and tissues. This astonishing transformation follows genetic programs, sequences of gene activation that unfold in precise order. Early genes divide the embryo into regions. Later genes specify what forms in each region. Still later, genes refine details, sculpting tissues and organs. Master control genes, like the Hox genes, are particularly important. They determine the body plan, specifying where the head goes, where limbs form, and how segments are arranged. Mutations in Hox genes can cause dramatic changes, like legs growing where antennae should be in fruit flies. These genes are ancient and remarkably conserved. The Hox genes that pattern a fly's body are related to those that pattern a human's. Development is robust, buffered against errors by redundancy and regulation, but it's not rigid. Environmental signals influence how genes are expressed. Nutrition, temperature, hormones, and even experiences can shape development. The genome provides the instructions, but the environment helps interpret them, ensuring that the organism fits its world. Genomes contain much more than just genes. For a long time, scientists focused on genes, the protein coding sequences, but genes make up less than 2% of the human genome. What's the rest? Much of it was once dismissed as junk DNA, thought to be evolutionary debris. But research is revealing that much of this non-coding DNA has function. Some non-coding regions regulate genes, acting as switches and dials that control when and where genes are expressed. Enhancers, regions that can be thousands of base pairs away from the genes they control, bind transcription factors and influence gene activity. Silencers do the opposite, repressing genes. The human genome contains millions of these regulatory elements. Other non-coding DNA produces RNA molecules that don't code for proteins, but have roles in gene regulation, chromosome structure, or RNA processing. Still other sequences are involved in the three-dimensional folding of chromosomes, bringing distant regions into contact so they can interact. And yes, some DNA may be junk, remnants of ancient viral infections, or duplicated genes that no longer function. But even junk can be useful. It provides raw material for evolution. Occasionally, a useless sequence mutates into something functional, creating new genes or regulatory elements. The genome is a library where even the discarded drafts might one day become stories. Transposons are mobile genetic elements. Scattered throughout your genome are sequences that can move. Transposons, or jumping genes, are DNA sequences capable of changing their position. Some cut themselves out and reinsert elsewhere. Others copy themselves, leaving the original in place while a new copy appears somewhere else. Nearly half of the human genome consists of transposons or their remnants. Most are ancient and no longer active, frozen in place by mutations. but some remain capable of jumping. When they do, they can disrupt genes, cause mutations, or alter gene regulation. Most such events are harmful or neutral, but occasionally a transposon insertion has beneficial effects. Transposons were discovered in corn by Barbara McClintock in the 1940s. Her work was so ahead of its time that it took decades for its importance to be recognized. She eventually received the Nobel Prize for revealing that genomes are dynamic, not static. Transposons drive evolution by creating genetic variation. They've shaped genomes across the tree of life. In some organisms, like fruit flies, transposons are active enough that each individual has a unique pattern of insertions. Controlling transposons is crucial for cells. Unchecked, they could wreak havoc on the genome. Cells use epigenetic silencing and other mechanisms to keep transposons in check. Genomes vary in size across species. The human genome contains about 3 billion base pairs. That sounds enormous, but it's actually modest compared to some organisms. The lungfish genome exceeds 40 billion base pairs. The Paris japonica plant has a genome over 150 billion base pairs, more than 50 times larger than ours. Genome size doesn't correlate with complexity. Single-celled amoebas can have genomes larger than mammals. The main factor determining genome size seems to be the accumulation of non-coding DNA, including transposons and duplicated regions. Species without efficient mechanisms to delete unnecessary DNA end up with bloated genomes. At the other extreme, some organisms have tiny genomes. Bacteria typically have a few million base pairs. Parasites, which can rely on their hosts for many functions, often have reduced genomes. The smallest known bacterial genome, from a symbiotic bacterium, is less than 200,000 base pairs. Genome size does affect cell size and metabolic rate. Organisms with large genomes tend to have larger cells and slower metabolism, possibly because more DNA takes longer to replicate. But the number of genes is more important than genome size for complexity. Humans have around 20,000 genes, similar to simpler organisms. Complexity arises from how genes are regulated and combined. Chromosome number varies widely too. Humans have 46 chromosomes, but this is not a universal standard. Dogs have 78, cats have 38, the fruit fly has just 8, the single-celled organism Paramecium has dozens. And at the extreme, the fern Ophioglossum reticulatum holds the record with more than 1,200 chromosomes. Chromosome number can change through evolution. Chromosomes can fuse, reducing the count, or split, increasing it. These changes are usually harmful, disrupting reproduction. But occasionally, a change establishes itself, becoming the new normal for a species. The difference between humans and great apes illustrates this. Humans have 46 chromosomes, while chimps, gorillas, and orangutans have 48. Human chromosome 2 is a fusion of 2 chromosomes that remain separate in other apes. The number of chromosomes doesn't determine the amount of genetic information. A species with many small chromosomes can have the same amount of DNA as a species with fewer large chromosomes. What matters is the total genome size and how genes are organized and regulated. Some organisms have more than 2 sets of chromosomes. Most animals, including humans, are diploid, having 2 sets of chromosomes, one from each parent. But some organisms are polyploid, with 3, 4, or even more sets. Polyploidy is common in plants. Wheat is hexaploid, with 6 sets of chromosomes. Strawberries are octoploid, with 8 sets. Polyploidy usually arises from errors in cell division or from hybridization between different species. An organism might fail to reduce chromosome number during reproduction, producing offspring with extra sets. Or two different species might interbreed, and their offspring might double their chromosomes, creating a new polyploid species. Polyploidy is usually lethal in animals, disrupting development, but it's tolerated in plants and has been a major force in plant evolution and agriculture. Many crop plants are polyploid, and the extra genetic material seems to provide advantages, like larger fruits or seeds. In animals, polyploidy is rare and usually restricted to certain tissues or developmental stages. Some cells, like liver cells, can become polyploid, and certain fish and amphibians are naturally polyploid, showing it's not impossible, just unusual, in the animal kingdom. Sex is determined by chromosomes in many species. In humans and most mammals, sex is determined by sex chromosomes. Females have 2 X chromosomes. Males have 1 X and 1 Y. The Y chromosome carries genes critical for male development, particularly the SRY gene, which triggers the development of testes. Without a functional Y chromosome, development follows the female pathway. This system, called XY sex determination, is common but not universal. Birds use a different system, ZW, where males have 2 Z chromosomes and females have 1 Z and 1 W. In some reptiles, sex isn't determined by chromosomes at all, but by temperature during egg incubation. Warmer temperatures produce one sex, cooler temperatures the other. Some organisms have no sex chromosomes. Sex is determined by a combination of genes on multiple chromosomes, or by environmental factors. And some organisms change sex during their lives, like certain fish that switch from female to male, or vice versa depending on social conditions. The diversity of sex determination systems shows that there's no single genetic blueprint for sex. Evolution has found many solutions to the challenge of producing 2 different sexes for reproduction. The X chromosome carries many important genes. The X chromosome is much larger than the Y and contains over 1,000 genes. Many of these genes have nothing to do with sex, but are essential for various functions. Because females have 2 X chromosomes and males have only 1, there's a potential imbalance. To prevent problems, females inactivate 1 X chromosome in each cell. This process, called X inactivation, randomly silences 1 X chromosome early in development. In some cells, the X from the mother is inactivated. In others, the X from the father. The choice is random and permanent for that cell and all its descendants. This creates a mosaic pattern. Female mammals are genetic mosaics, with some cells using 1 X chromosome and other cells using the other. A striking example is the calico cat. The gene for fur color is on the X chromosome. A female cat, with 1 X carrying the gene for orange fur and the other for black, will have patches of orange and black, the result of X inactivation creating a patchwork. Males with only 1 X can't be calico unless they have a rare chromosomal abnormality. X inactivation also affects X-linked genetic disorders. Females with 1 normal X and 1 mutated X are often mildly affected or unaffected because about half their cells use the normal X. Males with a mutated X have no backup, so they're fully affected. This is why conditions like colorblindness, hemophilia, and Duchenne muscular dystrophy are much more common in males. The Y chromosome is small but powerful. The Y chromosome is tiny compared to the X, carrying fewer than 100 genes. Over evolutionary time, it has lost most of its genes. The X and Y were once a matching pair, but the Y has degraded, shedding genes until only a small core remains. Some scientists once predicted the Y would disappear entirely, but it now appears to have stabilized. Despite its size, the Y chromosome has a critical role, determining male sex. The SRY gene triggers a cascade of developmental events that lead to male anatomy. Without SRY, even an embryo with a Y chromosome will develop as female, showing how essential this single gene is. The Y chromosome also carries genes important for sperm production. Mutations or deletions on the Y can cause male infertility. Because the Y chromosome doesn't recombine along most of its length, it's passed from father to son, essentially unchanged except for new mutations. This makes it useful for tracing paternal lineages through history. Some traits are linked to sex chromosomes. Traits controlled by genes on the X or Y chromosomes show unusual inheritance patterns. X-linked traits are more common in males because males have only one X. If a male inherits a recessive mutation on the X, he'll express the trait, having no second X to mask it. Colorblindness, particularly red-green colorblindness, is X-linked. About 8% of males are colorblind, but only about 0.5% of females. A female would need 2 copies of the mutation, one on each X, to be colorblind. Males need only one. Hemophilia, a disorder where blood doesn't clot properly, is also X-linked. Queen Victoria carried the mutation, and it spread through European royal families as her descendants married into various dynasties. The genetic history of royal hemophilia is well documented and illustrates how X-linked traits pass through families. Y-linked traits, controlled by genes on the Y chromosome, are rare because the Y has so few genes. Traits on the Y are passed from fathers to sons exclusively, never to daughters. Certain types of male infertility and some minor characteristics are Y-linked. Genetic recombination, shuffles, parental chromosomes. When sperm and egg cells form, chromosomes don't just segregate unchanged into the new cells. Homologous chromosomes, the matching pairs you inherited from your parents, first line up side by side. They then swap segments in a process called recombination or crossing over. This shuffling creates new combinations of alleles. Imagine chromosome 1 from your mother and chromosome 1 from your father. During recombination, they exchange segments. So the chromosome, one that goes into a sperm or egg, is a patchwork of both grandparents' chromosomes. This happens for all chromosome pairs during every round of sperm or egg production. Recombination is why siblings aren't identical. Even though they share the same parents, they receive different shuffled versions of the parental chromosomes. It's also why you're not a clone of any single grandparent. Each chromosome you carry is a unique blend. Recombination rates vary across the genome. Some regions recombine frequently, others rarely. Genes that are close together on a chromosome tend to be inherited together, a phenomenon called genetic linkage. But recombination can separate them, and the closer 2 genes are, the less likely recombination will occur between them. Linkage mapping locates genes on chromosomes. Before DNA sequencing became routine, scientists located genes by studying linkage. If 2 traits were usually inherited together, their genes were probably close together on the same chromosome. By analyzing inheritance patterns in families, researchers built linkage maps, showing the relative positions of genes. These maps were painstaking to create, requiring data from many families over multiple generations, but they were invaluable for finding disease genes. By tracking which chromosome regions were consistently inherited, along with a genetic disorder, scientists could narrow down the location of the responsible gene. The gene for Huntington's disease was located this way in the 1980 seconds, long before the genome was sequenced. Researchers analyzed large families affected by the disease, looking for genetic markers that segregated with the illness. They eventually pinned it to the short arm of chromosome 4. Years later, the precise mutation, an expansion of a CAG repeat in the HTT gene, was identified. Today, whole genome sequencing makes linkage mapping less necessary. We can now read the entire sequence and directly identify mutations. But linkage studies laid the groundwork for modern genetics and remain useful in certain situations. Population genetics studies, gene frequencies. Genetics isn't just about individuals, it's also about populations. Population genetics examines how gene frequencies change across groups of organisms over time. It combines genetics with evolution, asking questions about diversity, selection, drift, and migration. Within a population, multiple alleles of the same gene can coexist. Some might be common, others rare. The frequencies of these alleles can shift due to various forces. Natural selection favors alleles that enhance survival or reproduction, increasing their frequency. Genetic drift, random changes due to chance events, can also alter frequencies, especially in small populations. Migration introduces new alleles as individuals move between populations. Mutation continuously creates new alleles. The balance of these forces shapes genetic diversity within and between populations. High diversity means a population has many variants, which can be beneficial, providing raw material for adaptation. Low diversity can make a population vulnerable to disease or environmental change. Population genetics has practical applications. It helps conservationists manage endangered species by maintaining genetic diversity. It aids in understanding the spread of antibiotic resistance in bacteria. It informs studies of human evolution and migration, and it's the foundation for understanding how evolution works at the genetic level. The Hardy-Weinberg principle predicts allele frequencies. In population genetics, a key concept is the Hardy-Weinberg principle. It predicts that allele frequencies in a population will remain constant from generation to generation if certain conditions are met. No mutation, random mating, no selection, infinite population size, and no migration. Real populations never meet all these conditions, but the principle provides a baseline for detecting when evolution is occurring. If a population deviates from Hardy-Weinberg expectations, something is affecting allele frequencies. Selection might be favoring one allele, non-random mating might be occurring. Mutation might be introducing new variants. By measuring how far a population diverges from Hardy-Weinberg equilibrium, geneticists can infer what forces are at work. The principle also allows calculation of carrier frequencies for recessive disorders. If you know how common a disease is, you can estimate how many people are healthy carriers of the recessive allele. This is useful for genetic counseling and public health planning. Hardy-Weinberg is a mathematical tool, but it reveals something profound. In the absence of evolutionary forces, genetic variation is maintained. Rare alleles don't automatically disappear. They persist, sometimes for many generations, waiting for conditions where they might provide an advantage. Genetic drift has stronger effects in small populations. Genetic drift is the random change in allele frequencies due to chance. In large populations, drift is weak because random events average out. But in small populations, drift can be powerful, causing allele frequencies to change dramatically, even without selection. Imagine a small population with two alleles at equal frequency. Just by chance, more individuals carrying one allele might reproduce, shifting the balance. Over generations, one allele might become common or even fixed, while the other disappears, not because it was better or worse, but purely by luck. Bottlenecks, where a population shrinks to a small size, amplify drift. The survivors carry only a subset of the original genetic diversity, and their alleles, by chance, become the foundation of the recovered population. Founder effects, where a few individuals establish a new population, similarly reduce diversity. These effects are important in conservation. Small or fragmented populations lose genetic diversity, making them less able to adapt. Inbreeding becomes more likely, increasing the risk of harmful recessive traits appearing. Maintaining large, connected populations preserves diversity and resilience. Inbreeding increases risks of genetic disorders. When close relatives reproduce, their offspring are more likely to inherit two copies of the same allele, including harmful recessive alleles. This is inbreeding, and it can have serious consequences. Inbred individuals have increased risk of genetic disorders, reduced fertility, and lower survival rates, collectively known as inbreeding depression. Many harmful recessive alleles are rare in populations. The chance that two unrelated people both carry the same rare recessive allele is small. But if they're relatives, they're more likely to have inherited the same allele from a common ancestor. Their children then have a higher chance of inheriting two copies. Historically, royal families and isolated communities practiced close inbreeding, leading to health problems. The Habsburgs, European royalty who intermarried for generations, exhibited numerous genetic disorders, likely due to inbreeding. Island populations, or small isolated groups, often show higher rates of certain genetic conditions for the same reason. Most cultures have taboos against close inbreeding, likely because the negative effects were observed long before genetics was understood. Modern genetic counseling advises against reproduction between close relatives, precisely because of these increased risks. Genetic diversity is crucial for species survival. A genetically diverse population is a healthy population. Diversity provides options. When the environment changes, whether through climate shift, new diseases, or other pressures, a diverse population is more likely to include individuals with traits that allow survival. Those individuals reproduce, passing on their genes, and the population adapts. In contrast, a population with low diversity might lack the variants needed to survive new challenges. If all individuals are genetically similar, a disease that affects one could affect all. An environmental shift that harms one could harm all. The population might decline or even go extinct. Conservation biologists work to preserve genetic diversity in endangered species. They manage breeding programs to minimize inbreeding and maximize genetic variation. They connect fragmented populations to allow gene flow. They sometimes even introduce individuals from other populations to boost diversity. Humans are relatively low in genetic diversity compared to many species, a consequence of our evolutionary history and past population bottlenecks. But the diversity we have is precious, representing adaptations to diverse environments across the globe. Preserving human genetic diversity means maintaining the full breadth of our species' evolutionary heritage. Genetic bottlenecks reduce diversity dramatically. A genetic bottleneck occurs when a population's size shrinks dramatically, whether due to disaster, disease, or geographic isolation. The survivors carry only a fraction of the original genetic diversity. When the population recovers, it rebuilds from this limited gene pool. Cheetahs are an example of an ancient bottleneck. All living cheetahs are remarkably genetically similar, suggesting that at some point in the past, their population dropped to very few individuals. This low diversity has consequences. Cheetahs have high rates of reproductive problems and are more vulnerable to diseases. Northern elephant seals also experienced a bottleneck. Hunted nearly to extinction in the 19th century, the population dropped to perhaps 20 individuals. They've since recovered to over 100,000, but genetic studies show remarkably little diversity. All living elephant seals are descended from that tiny group of survivors. Humans also experienced bottlenecks in our evolutionary past. Some geneticists believe that at one time, the human population may have dropped to as few as a few thousand individuals. We carry the genetic signature of that near extinction in our relatively low diversity compared to other species. Founder effects shape island populations. When a small group of individuals colonizes a new area, they carry only a subset of the genetic variation from their source population. This founder effect can create populations with unusual allele frequencies. By chance, certain alleles might be common among the founders, even if they were rare in the original population. Island populations often show founder effects. A few individuals arrive on an island, and all subsequent inhabitants descend from them. If one founder carried a rare allele, that allele might become common on the island, even if it's rare elsewhere. Over time, the island population becomes genetically distinct. The Amish populations in North America show founder effects. They're descended from a small number of founders who emigrated from Europe. Certain rare genetic disorders are more common among the Amish than in the general population because, by chance, the founders carried those alleles. Founder effects can be seen in human history whenever populations migrated to new areas. Each migration involved a subset of people carrying a subset of genetic variation. As humans spread across the globe, successive founder effects contributed to the genetic differences we see between populations today. Migration and gene flow connect populations. Migration, the movement of individuals between populations, is a powerful force in genetics. When individuals migrate and reproduce, they introduce new alleles into the population, increasing diversity and preventing populations from diverging too much. This gene flow counteracts the effects of drift and can spread beneficial alleles across wide areas. Gene flow has been a constant in human history. People have always moved, whether for trade, war, exploration, or survival. These movements mixed genetic variants, preventing human populations from becoming separate species. Despite geographic separation and adaptation to different environments, all humans can interbreed and produce fertile offspring. We remain one species, connected by gene flow. Today, migration continues to shape human genetic diversity. As people move and form families across traditional boundaries, genetic mixing increases. This blending enriches the human gene pool and underscores our shared heritage. We are, at a genetic level, one global family. In conservation, maintaining gene flow between fragmented populations is crucial. Corridors that allow animals to move between habitat patches prevent isolated populations from inbreeding and losing diversity. Gene flow sustains genetic health across landscapes. Natural selection favors beneficial alleles. Natural selection is the process by which alleles that enhance survival or reproduction become more common over generations. If an allele helps an organism avoid predators, find food, resist disease, or attract mates, individuals carrying that allele are more likely to pass it on. Over time, the allele spreads through the population. Selection can act on existing variation or on new mutations. A mutation that improves an enzyme's efficiency or changes a protein's structure in a beneficial way will be favored. Even a small advantage, if consistent, can drive an allele from rarity to commonality over many generations. Not all selection is positive. Negative selection removes harmful alleles, preventing them from spreading. Most new mutations are neutral or slightly harmful, and selection weeds them out. This purifying selection maintains the functionality of genes, preventing the accumulation of damaging changes. Balancing selection maintains multiple alleles in a population because diversity itself is advantageous. The sickle cell example is classic. Heterozygotes, with one normal and one sickle cell allele, have protection against malaria without the severe symptoms of sickle cell disease. In malaria-prone regions, this balance keeps both alleles common. Some alleles persist due to heterozygote advantage. Heterozygote advantage, where having two different alleles is better than having two copies of the same allele, is a form of balancing selection. The sickle cell trait is the best-known example, but there are others. Certain MHC genes, important for immune function, show heterozygote advantage. Having diverse MHC alleles allows the immune system to recognize a wider range of pathogens. In populations where heterozygote advantage exists, both alleles persist, even if one is harmful in the homozygous state. The advantage to heterozygotes outweighs the cost to homozygotes, maintaining both alleles at intermediate frequencies. This phenomenon complicates disease genetics. An allele that seems harmful might persist because it provides benefits in certain contexts or combinations. Understanding these nuances is important for predicting disease risk and for genetic counseling. Heterozygote advantage also illustrates that fitness is context-dependent. An allele that's beneficial in one environment might be neutral or harmful in another. There's no universally best allele, only alleles suited to particular circumstances. Genetic load refers to harmful mutations we carry. Every person carries some harmful mutations. These are part of the genetic load, the burden of deleterious alleles present in a population. Most are recessive and cause no problems unless paired with another copy. Others are mildly harmful, having small effects that don't completely prevent reproduction. The genetic load exists because mutation is constant and selection is not perfect. New mutations arise faster than selection can remove them, especially if they're rare or have small effects. In large populations, genetic load is spread across many individuals. In small populations, it can concentrate, increasing the likelihood that harmful alleles appear in homozygous form. Genetic load is one reason why inbreeding is harmful. It increases the chance that two copies of a deleterious recessive allele come together, turning a hidden load into visible problems. Outbreeding, reproducing with unrelated individuals, helps mask the genetic load by ensuring diverse allele combinations. Understanding genetic load has implications for medicine and public health. It suggests that everyone carries risk alleles, not just those with family histories of disease. As we identify more of these alleles through genomic studies, we gain a fuller picture of human health and disease risk. Genetic counselors help families navigate genetic risks. Genetic counselors are trained professionals who help individuals and families understand genetic information. They assess risk for inherited conditions, explain test results, and support decision-making. If you're considering genetic testing, planning a family, or have a family history of genetic disease, a genetic counselor can provide guidance. Counseling often begins with taking a detailed family history, constructing a pedigree that shows which relatives have been affected by health conditions. This visual tool helps identify patterns of inheritance. The counselor assesses risk based on the pedigree, known genetics, and sometimes genetic testing. If testing is recommended, the counselor explains what the test can and can't reveal, the meaning of different results, and the implications for the individual and family. After testing, they help interpret results, which can be complex. A positive result might indicate high risk but not certainty. A negative result might be reassuring but doesn't eliminate all risk. Genetic counselors also provide emotional support. Learning about genetic risks can be stressful. Decisions about testing, treatment, or family planning can be difficult. Counselors help families navigate these challenges with empathy and expertise. respecting their autonomy while providing information. Prenatal genetic testing detects conditions before birth. Advances in genetics have made it possible to test fetuses for genetic conditions during pregnancy. Prenatal testing can identify chromosomal abnormalities like Down syndrome or genetic disorders like cystic fibrosis, allowing parents to prepare or make informed decisions. Non-invasive prenatal testing analyzes fetal DNA present in the mother's blood. It's safe and can screen for certain chromosomal conditions early in pregnancy. More invasive tests, like amniocentesis or chorionic villus sampling, involve taking samples of amniotic fluid or placental tissue. These carry small risks but provide more comprehensive genetic information. Prenatal testing raises ethical questions. What should parents do with the information? Some choose to continue pregnancies regardless of results, using the knowledge to prepare. Others face difficult decisions about termination. Different families have different values, and genetic counselors help them explore options without judgment. As prenatal testing becomes more advanced, it's increasingly able to detect a wider range of conditions, including milder ones or predispositions to late-onset diseases. This raises questions about where to draw lines and how much information is helpful versus overwhelming. Newborn screening identifies treatable genetic conditions. Shortly after birth, most babies undergo newborn screening, a blood test that checks for dozens of genetic and metabolic disorders. These conditions, if detected early, can often be managed with diet, medication, or other interventions, preventing serious health problems. Phenylketonuria, or PKU, is one example. Babies with PKU can't properly break down an amino acid called phenylalanine. Without treatment, it builds up and damages the brain, causing intellectual disability. But if detected through newborn screening, a special diet can prevent these effects. Children with PKU who follow the diet develop normally. Other conditions screened for include sickle cell disease, congenital hypothyroidism, and cystic fibrosis. Early detection and treatment save lives and prevent disability. Newborn screening is a major public health success, protecting millions of children. As genetic knowledge grows, the list of conditions included in newborn screening expands. Some regions test for more than 60 disorders. The challenge is balancing the benefit of early detection with the burden of false positives, unnecessary worry, and the cost of expanded screening. Pharmacogenomics personalizes medicine based on genes. People respond differently to medications. A drug that works well for one person might be ineffective or cause side effects in another. Some of this variation is due to genetics. Genes influence how drugs are metabolized, how they interact with targets in the body, and how the body responds to them. Pharmacogenomics studies how genetic variation affects drug response. By testing for specific gene variants, doctors can predict which medications are likely to work best for a patient and at what dose. This personalized approach can improve treatment outcomes and reduce adverse reactions. One example is the CYP450 genes, which code for enzymes that break down many drugs. Variants in these genes can make someone a rapid metabolizer who breaks down a drug quickly and might need higher doses, or a poor metabolizer who processes the drug slowly and might need lower doses or different medications. Pharmacogenomics is increasingly used in psychiatry, oncology, and other fields. Cancer treatments can be tailored based on genetic mutations in the tumor. Antidepressant choice can be informed by metabolic gene variants. As our understanding grows, medicine is moving toward treatments tailored to each person's genetic makeup. Cancer often results from accumulated genetic mutations. Cancer is, at its core, a genetic disease. It arises when cells accumulate mutations that cause them to grow and divide uncontrollably. These mutations usually affect genes that regulate cell division, DNA repair, or cell death. When these controls fail, a cell can become cancerous. Most cancers require multiple mutations. A single hit usually isn't enough. Cells have many safeguards, and several must fail before cancer develops. This is why cancer incidence increases with age. Over a lifetime, cells divide many times, each division a chance for errors. Mutations accumulate, eventually reaching a threshold where cancer emerges. Some people inherit mutations that predispose them to cancer. BRCA1 and BRCA2 mutations increase breast and ovarian cancer risk. Lynch syndrome involves mutations in DNA repair genes, raising the risk of colon and other cancers. Inheriting such a mutation doesn't guarantee cancer, but it increases vulnerability because one safeguard is already compromised. Environmental factors also cause mutations. Tobacco smoke, UV radiation, certain chemicals, and viruses can damage DNA, increasing cancer risk. Cancer is a convergence of genetic susceptibility and environmental exposure, a disease of both nature and nurture. Tumor DNA reveals mutations driving cancer. When a tumor forms, it carries mutations that distinguish its DNA from normal cells. Sequencing tumor DNA can reveal which genes are mutated, providing insights into how the cancer developed and how it might be treated. Some mutations activate oncogenes, genes that promote cell growth. Others inactivate tumor suppressor genes, which normally restrain growth. Different cancers have different mutational signatures. Lung cancers in smokers often have mutations caused by tobacco carcinogens. Skin cancers in sun-exposed areas show mutations caused by UV light. These signatures are like fingerprints, revealing the history of the tumor. Understanding a tumor's genetic makeup allows targeted therapies. Some drugs are designed to specifically attack cancer cells with particular mutations. For example, certain lung cancers with mutations in the EGFR gene can be treated with EGFR inhibitors. Patients whose tumors have these mutations respond well, while those without them don't benefit. Precision oncology, using genetic information to guide cancer treatment, is transforming care. By matching treatments to the genetic profile of each patient's tumor, doctors can improve outcomes and reduce unnecessary side effects from ineffective therapies. Population genetics studies, gene frequencies. Genetics isn't just about individuals, it's also about populations. Population genetics examines how gene frequencies change across groups of organisms over time. It combines genetics with evolution, asking questions about diversity, selection, drift, and migration. Within a population, multiple alleles of the same gene can coexist. Some might be common, others rare. The frequencies of these alleles can shift due to various forces. Natural selection favors alleles that enhance survival or reproduction, increasing their frequency. Genetic drift, random changes due to chance events, can also alter frequencies, especially in small populations. Migration introduces new alleles as individuals move between populations. Mutation continuously creates new alleles. The balance of these forces shapes genetic diversity within and between populations. High diversity means a population has many variants, which can be beneficial, providing raw material for adaptation. Low diversity can make a population vulnerable to disease or environmental change. Population genetics has practical applications. It helps conservationists manage endangered species by maintaining genetic diversity. It aids in understanding the spread of antibiotic resistance in bacteria. It informs studies of human evolution and migration, and it's the foundation for understanding how evolution works at the genetic level. The Hardy-Weinberg principle predicts allele frequencies. In population genetics, a key concept is the Hardy-Weinberg principle. It predicts that allele frequencies in a population will remain constant from generation to generation if certain conditions are met. No mutation, random mating, no selection, infinite population size, and no migration. Real populations never meet all these conditions, but the principle provides a baseline for detecting when evolution is occurring. If a population deviates from Hardy-Weinberg expectations, something is affecting allele frequencies. Selection might be favoring one allele. Non-random mating might be occurring. Mutation might be introducing new variants. By measuring how far a population diverges from Hardy-Weinberg equilibrium, geneticists can infer what forces are at work. The principle also allows calculation of carrier frequencies for recessive disorders. If you know how common a disease is, you can estimate how many people are healthy carriers of the recessive allele. This is useful for genetic counseling and public health planning. Hardy-Weinberg is a mathematical tool, but it reveals something profound. In the absence of evolutionary forces, genetic variation is maintained. Rare alleles don't automatically disappear. They persist, sometimes for many generations, waiting for conditions where they might provide an advantage. Genetic drift has stronger effects. In small populations, genetic drift is the random change in allele frequencies due to chance. In large populations, drift is weak because random events average out. But in small populations, drift can be powerful, causing allele frequencies to change dramatically, even without selection. Imagine a small population with two alleles at equal frequency. Just by chance, more individuals carrying one allele might reproduce, shifting the balance. Over generations, one allele might become common or even fixed, while the other disappears, not because it was better or worse, but purely by luck. Bottlenecks, where a population shrinks to a small size, amplify drift. The survivors carry only a subset of the original genetic diversity, and their alleles, by chance, become the foundation of the recovered population. Founder effects, where a few individuals establish a new population, similarly reduce diversity. These effects are important in conservation. Small or fragmented populations lose genetic diversity, making them less able to adapt. Inbreeding becomes more likely, increasing the risk of harmful recessive traits appearing. Maintaining large, connected populations preserves diversity and resilience. Inbreeding increases risks of genetic disorders. When close relatives reproduce, their offspring are more likely to inherit two copies of the same allele, including harmful recessive alleles. This is inbreeding, and it can have serious consequences. Inbred individuals have increased risk of genetic disorders, reduced fertility, and lower survival rates, collectively known as inbreeding depression. Many harmful recessive alleles are rare in populations. The chance that two unrelated people both carry the same rare recessive allele is small. But if they're relatives, they're more likely to have inherited the same allele from a common ancestor. Their children then have a higher chance of inheriting two copies. Historically, royal families and isolated communities practiced close inbreeding, leading to health problems. The Habsburgs, European royalty who intermarried for generations, exhibited numerous genetic disorders, likely due to inbreeding. Island populations, or small isolated groups, often show higher rates of certain genetic conditions for the same reason. Most cultures have taboos against close inbreeding, likely because the negative effects were observed long before genetics was understood. Modern genetic counseling advises against reproduction between close relatives, precisely because of these increased risks. Genetic diversity is crucial for species survival. A genetically diverse population is a healthy population. Diversity provides options. When the environment changes, whether through climate shift, new diseases, or other pressures, a diverse population is more likely to include individuals with traits that allow survival. Those individuals reproduce, passing on their genes, and the population adapts. In contrast, a population with low diversity might lack the variance needed to survive new challenges. If all individuals are genetically similar, a disease that affects one could affect all. An environmental shift that harms one could harm all. The population might decline or even go extinct. Conservation biologists work to preserve genetic diversity in endangered species. They manage breeding programs to minimize inbreeding and maximize genetic variation. They connect fragmented populations to allow gene flow. They sometimes even introduce individuals from other populations to boost diversity. Humans are relatively low in genetic diversity compared to many species, a consequence of our evolutionary history and past population bottlenecks. But the diversity we have is precious, representing adaptations to diverse environments across the globe. Preserving human genetic diversity means maintaining the full breadth of our species' evolutionary heritage. Genetic bottlenecks reduce diversity dramatically. A genetic bottleneck occurs when a population's size shrinks dramatically, whether due to disaster, disease, or geographic isolation. The survivors carry only a fraction of the original genetic diversity. When the population recovers, it rebuilds from this limited gene pool. Cheetahs are an example of an ancient bottleneck. All living cheetahs are remarkably genetically similar, suggesting that at some point in the past, their population dropped to very few individuals. This low diversity has consequences. Cheetahs have high rates of reproductive problems and are more vulnerable to diseases. Northern elephant seals also experienced a bottleneck. Hunted nearly to extinction in the 19th century, the population dropped to perhaps 20 individuals. They've since recovered to over 100,000, but genetic studies show remarkably little diversity. All living elephant seals are descended from that tiny group of survivors. Humans also experienced bottlenecks in our evolutionary past. Some geneticists believe that at one time, the human population may have dropped to as few as a few thousand individuals. We carry the genetic signature of that near extinction in our relatively low diversity compared to other species. Founder effects shape island populations. When a small group of individuals colonizes a new area, they carry only a subset of the genetic variation from their source population. This founder effect can create populations with unusual allele frequencies. By chance, certain alleles might be common among the founders even if they were rare in the original population. Island populations often show founder effects. A few individuals arrive on an island, and all subsequent inhabitants descend from them. If one founder carried a rare allele, that allele might become common on the island, even if it's rare elsewhere. Over time, the island population becomes genetically distinct. The Amish populations in North America show founder effects. They're descended from a small number of founders who emigrated from Europe. Certain rare genetic disorders are more common among the Amish than in the general population because, by chance, the founders carried those alleles. Founder effects can be seen in human history whenever populations migrated to new areas. Each migration involved a subset of people carrying a subset of genetic variation. As humans spread across the globe, successive founder effects contributed to the genetic differences we see between populations today. Migration and gene flow connect populations. Migration, the movement of individuals between populations, is a powerful force in genetics. When individuals migrate and reproduce, they introduce new alleles into the population, increasing diversity and preventing populations from diverging too much. This gene flow counteracts the effects of drift and can spread beneficial alleles across wide areas. Gene flow has been a constant in human history. People have always moved, whether for trade, war, exploration, or survival. These movements mixed genetic variants, preventing human populations from becoming separate species. Despite geographic separation and adaptation to different environments, all humans can interbreed and produce fertile offspring. We remain one species, connected by gene flow. Today, migration continues to shape human genetic diversity. As people move and form families across traditional boundaries, genetic mixing increases. This blending enriches the human gene pool and underscores our shared heritage. We are, at a genetic level, one global family. In conservation, maintaining gene flow between fragmented populations is crucial. Corridors that allow animals to move between habitat patches prevent isolated populations from inbreeding and losing diversity. Gene flow sustains genetic health across landscapes. Natural selection favors beneficial alleles. Natural selection is the process by which alleles that enhance survival or reproduction become more common over generations. If an allele helps an organism avoid predators, find food, resist disease, or attract mates, individuals carrying that allele are more likely to pass it on. Over time, the allele spreads through the population. Selection can act on existing variation or on new mutations. A mutation that improves an enzyme's efficiency or changes a protein's structure in a beneficial way will be favored. Even a small advantage, if consistent, can drive an allele from rarity to commonality over many generations. Not all selection is positive. Negative selection removes harmful alleles, preventing them from spreading. Most new mutations are neutral or slightly harmful, and selection weeds them out. This purifying selection maintains the functionality of genes, preventing the accumulation of damaging changes. Balancing selection maintains multiple alleles in a population because diversity itself is advantageous. The sickle cell example is classic. Heterozygotes with one normal and one sickle cell allele have protection against malaria without the severe symptoms of sickle cell disease. In malaria-prone regions, this balance keeps both alleles common. Some alleles persist due to heterozygote advantage. Heterozygote advantage, where having two different alleles is better than having two copies of the same allele, is a form of balancing selection. The sickle cell trait is the best-known example, but there are others. Certain MHC genes, important for immune function, show heterozygote advantage. Having diverse MHC alleles allows the immune system to recognize a wider range of pathogens. In populations where heterozygote advantage exists, both alleles persist, even if one is harmful in the homozygous state. The advantage to heterozygotes outweighs the cost to homozygotes, maintaining both alleles at intermediate frequencies. This phenomenon complicates disease genetics. An allele that seems harmful might persist because it provides benefits in certain contexts or combinations. Understanding these nuances is important for predicting disease risk and for genetic counseling. Heterozygote advantage also illustrates that fitness is context-dependent. An allele that's beneficial in one environment might be neutral or harmful in another. There's no universally best allele, only alleles suited to particular circumstances. Genetic load refers to harmful mutations we carry. Every person carries some harmful mutations. These are part of the genetic load, the burden of deleterious alleles present in a population. Most are recessive and cause no problems unless paired with another copy. Others are mildly harmful, having small effects that don't completely prevent reproduction. The genetic load exists because mutation is constant and selection is not perfect. New mutations arise faster than selection can remove them, especially if they're rare or have small effects. In large populations, genetic load is spread across many individuals. In small populations, it can concentrate, increasing the likelihood that harmful alleles appear in homozygous form. Genetic load is one reason why inbreeding is harmful. It increases the chance that two copies of a deleterious recessive allele come together, turning a hidden load into visible problems. Outbreeding, reproducing with unrelated individuals, helps mask the genetic load by ensuring diverse allele combinations. Understanding genetic load has implications for medicine and public health. It suggests that everyone carries risk alleles, not just those with family histories of disease. As we identify more of these alleles through genomic studies, we gain a fuller picture of human health and disease risk. Genetic counselors help families navigate genetic risks. Genetic counselors are trained professionals who help individuals and families understand genetic information. They assess risk for inherited conditions, explain test results, and support decision-making. If you're considering genetic testing, planning a family, or have a family history of genetic disease, a genetic counselor can provide guidance. Counseling often begins with taking a detailed family history, constructing a pedigree that shows which relatives have been affected by health conditions. This visual tool helps identify patterns of inheritance. The counselor assesses risk based on the pedigree, known genetics, and sometimes genetic testing. If testing is recommended, the counselor explains what the test can and can't reveal, the meaning of different results, and the implications for the individual and family. After testing, they help interpret results, which can be complex. A positive result might indicate high risk but not certainty. A negative result might be reassuring but doesn't eliminate all risk. Genetic counselors also provide emotional support. Learning about genetic risks can be stressful. Decisions about testing, treatment, or family planning can be difficult. Counselors help families navigate these challenges with empathy and expertise, respecting their autonomy while providing information. Prenatal genetic testing detects conditions before birth. Advances in genetics have made it possible to test fetuses for genetic conditions during pregnancy. Prenatal testing can identify chromosomal abnormalities like Down syndrome or genetic disorders like cystic fibrosis, allowing parents to prepare or make informed decisions. Non-invasive prenatal testing analyzes fetal DNA present in the mother's blood. It's safe and can screen for certain chromosomal conditions early in pregnancy. More invasive tests, like amniocentesis or chorionic villus sampling, involve taking samples of amniotic fluid or placental tissue. These carry small risks, but provide more comprehensive genetic information. Prenatal testing raises ethical questions. What should parents do with the information? Some choose to continue pregnancies regardless of results, using the knowledge to prepare. Others face difficult decisions about termination. Different families have different values, and genetic counselors help them explore options without judgment. As prenatal testing becomes more advanced, it's increasingly able to detect a wider range of conditions, including milder ones or predispositions to late-onset diseases. This raises questions about where to draw lines and how much information is helpful versus overwhelming. Newborn screening identifies treatable genetic conditions. Shortly after birth, most babies undergo newborn screening, a blood test that checks for dozens of genetic and metabolic disorders. These conditions, if detected early, can often be managed with diet, medication, or other interventions, preventing serious health problems. Phenylketonuria, or PKU, is one example. Babies with PKU can't properly break down an amino acid called phenylalanine. Without treatment, it builds up and damages the brain, causing intellectual disability. But if detected through newborn screening, a special diet can prevent these effects. Children with PKU who follow the diet develop normally. Other conditions screened for include sickle cell disease, congenital hypothyroidism, and cystic fibrosis. Early detection and treatment save lives and prevent disability. Newborn screening is a major public health success, protecting millions of children. As genetic knowledge grows, the list of conditions included in newborn screening expands. Some regions test for more than 60 disorders. The challenge is balancing the benefit of early detection with the burden of false positives, unnecessary worry, and the cost of expanded screening. Pharmacogenomics personalizes medicine based on genes. People respond differently to medications. A drug that works well for one person might be ineffective or cause side effects in another. Some of this variation is due to genetics. Genes influence how drugs are metabolized, how they interact with targets in the body, and how the body responds to them. Pharmacogenomics studies how genetic variation affects drug response. By testing for specific gene variants, doctors can predict which medications are likely to work best for a patient and at what dose. This personalized approach can improve treatment outcomes and reduce adverse reactions. One example is the CYP450 genes, which code for enzymes that break down many drugs. Variants in these genes can make someone a rapid metabolizer who breaks down a drug quickly and might need higher doses, or a poor metabolizer who processes the drug slowly and might need lower doses or different medications. Pharmacogenomics is increasingly used in psychiatry, oncology, and other fields. Cancer treatments can be tailored based on genetic mutations in the tumor. Antidepressant choice can be informed by metabolic gene variants. As our understanding grows, medicine is moving toward treatments tailored to each person's genetic makeup. Cancer often results from accumulated genetic mutations. Cancer is, at its core, a genetic disease. It arises when cells accumulate mutations that cause them to grow and divide uncontrollably. These mutations usually affect genes that regulate cell division, DNA repair, or cell death. When these controls fail, a cell can become cancerous. Most cancers require multiple mutations. A single hit usually isn't enough. Cells have many safeguards, and several must fail before cancer develops. This is why cancer incidence increases with age. Over a lifetime, cells divide many times, each division a chance for errors. Mutations accumulate, eventually reaching a threshold where cancer emerges. Some people inherit mutations that predispose them to cancer. BRCA1 and BRCA2 mutations increase breast and ovarian cancer risk. Lynch syndrome involves mutations in DNA repair genes, raising the risk of colon and other cancers. Inheriting such a mutation doesn't guarantee cancer, but it increases vulnerability because one safeguard is already compromised. Environmental factors also cause mutations. Tobacco smoke, UV radiation, certain chemicals, and viruses can damage DNA, increasing cancer risk. Cancer is a convergence of genetic susceptibility and environmental exposure, a disease of both nature and nurture. Tumor DNA reveals mutations, driving cancer. When a tumor forms, it carries mutations that distinguish its DNA from normal cells. Sequencing tumor DNA can reveal which genes are mutated, providing insights into how the cancer developed and how it might be treated. Some mutations activate oncogenes, genes that promote cell growth. Others inactivate tumor suppressor genes, which normally restrain growth. Different cancers have different mutational signatures. Lung cancers in smokers often have mutations caused by tobacco carcinogens. Skin cancers in sun-exposed areas show mutations caused by UV light. These signatures are like fingerprints, revealing the history of the tumor. Understanding a tumor's genetic makeup allows targeted therapies. Some drugs are designed to specifically attack cancer cells with particular mutations. For example, certain lung cancers with mutations in the EGFR gene can be treated with EGFR inhibitors. Patients whose tumors have these mutations respond well, while those without them don't benefit. Precision oncology, using genetic information to guide cancer treatment, is transforming care. By matching treatments to the genetic profile of each patient's tumor, doctors can improve outcomes and reduce unnecessary side effects from ineffective therapies. Forensic genetics solves crimes and identifies remains. DNA is unique to each individual, making it a powerful forensic tool. DNA found at crime scenes, on clothing, or on weapons can link suspects to crimes. DNA profiling examines specific variable regions of the genome, creating a genetic fingerprint. Forensic DNA analysis has exonerated wrongly convicted individuals and identified perpetrators years after crimes. Cold cases have been solved using DNA evidence that couldn't be analyzed with older techniques. The technology has also identified victims of disasters when other methods fail. Genetic genealogy, using DNA databases to find relatives, has become a tool for law enforcement. By comparing crime scene DNA to databases of people who've taken ancestry tests, investigators identify suspects through family connections. This approach solved notorious cases, but also raises privacy concerns, as it involves genetic information from people who haven't consented to being in a criminal database. Balancing justice with privacy is challenging. DNA evidence is powerful, but not infallible. Contamination, errors, and statistical misinterpretation can occur. And the use of genetic databases for law enforcement, while effective, creates tensions between public safety and individual rights. Paternity testing confirms biological relationships. DNA testing can determine biological relationships with high accuracy. Paternity tests compare a child's DNA to a potential father's, looking for shared genetic markers. Because a child inherits half their DNA from each parent, a biological father will share specific patterns with the child. These tests are used in legal cases, immigration applications, and personal situations. They can resolve disputes, establish parental rights, or confirm family connections. The accuracy is extremely high, with probabilities exceeding 99.9% when there's a match. Beyond paternity, DNA testing can identify other relationships. Sibling tests compare DNA to determine if two people share one or both parents. Grandparentage tests can establish connections when a parent isn't available for testing. These tests rely on the fact that relatives share more DNA than unrelated individuals, with closer relatives sharing more. The ease of DNA testing has made it common, but it can also reveal unexpected truths. Non-paternity, adoptions, or unknown siblings might be discovered. These revelations can be emotional, affecting families in complex ways. Genetic information is powerful, and with it comes responsibility for sensitivity and care. Ancestry testing traces your genetic heritage. Ancestry DNA tests analyze your genome to estimate your ethnic background and identify relatives. By comparing your DNA to reference populations from around the world, companies estimate what percentage of your ancestry comes from different regions. These estimates are based on patterns of genetic variation, Populations that have been geographically isolated accumulate specific variants. If you carry variants common in a particular region, you likely have ancestry from there. The more data companies have, the more refined their estimates become. Ancestry tests can also find relatives. By comparing your DNA to others in the database, they identify people who share significant amounts of DNA with you, indicating common ancestors. Many people have reconnected with distant relatives or discovered unknown family branches this way. But ancestry estimates have limitations. They're probabilities, not certainties. Ethnic categories are social constructs, not strict biological divisions. And ancient human migrations and mixing mean that ancestry is complex. Still, ancestry testing offers a window into your genetic past, connecting you to the global story of human movement and diversity. Mitochondrial DNA traces deep maternal lineages. Because mitochondrial DNA is inherited maternally without recombination, it's ideal for tracing deep ancestry. Your mtDNA is virtually identical to your mother's and her mother's, stretching back thousands of generations. Mutations accumulate slowly, creating lineages that can be traced across continents and millennia. By comparing your mtDNA to databases of known lineages, ancestry tests can trace your maternal haplogroup and the path your maternal ancestors took as humans spread from Africa. You might learn that your maternal line passed through the Middle East during the Neolithic, or crossed into Europe during the Ice Age, or arrived in the Americas thousands of years ago. These deep ancestry stories are population-level narratives. They tell the story of groups, not individuals. But knowing your haplogroup connects you to a broader human heritage, to migrations and movements that shaped our species. Mitochondrial DNA has been used to study ancient populations, comparing DNA from modern people to that from archaeological remains. It's revealed connections between ancient and modern groups, migration routes, and the timing of key events in human prehistory. Y chromosome analysis reveals paternal lineages. For males, Y chromosome analysis provides a patrilineal counterpart to mitochondrial DNA. Your Y chromosome is nearly identical to your father's and his father's, stretching back through generations. Like mtDNA, the Y chromosome accumulates mutations that create haplogroups, marking different lineages. U chromosome testing can trace the paths of paternal ancestors, revealing migrations and population movements from a male perspective. Historical events, like the spread of agriculture or the expansion of empires, left genetic marks on Y chromosomes that can still be detected. Famous examples include the Genghis Khan Y chromosome lineage. A specific Y chromosome haplogroup is extremely common in Central and East Asia, and genetic analysis suggests it spread from a single ancestor about 1,000 years ago, during Genghis Khan's reign. While it can't be proven that Khan himself is the ancestor, the timing and geography fit, and it illustrates how one individual's genetic legacy can be remarkably widespread. Y chromosome studies have also revealed the African origins of all human males, tracing back to Y chromosomal Adam. Like mitochondrial Eve, he's the most recent common patrilineal ancestor of all living men, representing a node in the vast family tree of humanity. DNA can survive for thousands of years in the right conditions. DNA is fragile. Heat, moisture, and oxygen degrade it. In most environments, DNA decays within decades or centuries. But in cold, dry, or oxygen-poor conditions, it can persist much longer. Permafrost, caves, and dry deserts preserve DNA remarkably well. The oldest DNA recovered so far comes from mammoth teeth frozen in Siberian permafrost for over 1 million years. While highly fragmented, enough remains to sequence and analyze. Most ancient DNA is younger, thousands to tens of thousands of years old, but still provides valuable insights. Ancient human DNA has been extracted from bones and teeth found in archaeological sites. It's revealed the genetic makeup of past populations, migrations, and interactions between groups like Neanderthals and modern humans. Ancient DNA from animals and plants illuminates ecology and evolution in the past. Extracting and analyzing ancient DNA is technically challenging. Contamination with modern DNA is a constant risk. The DNA is damaged and fragmented, requiring specialized techniques to sequence and assemble. But advances in technology have made ancient DNA a powerful tool for understanding the past. Ancient DNA reveals Neanderthal interbreeding. One of the most exciting discoveries from ancient DNA is that modern humans interbred with Neanderthals. By sequencing Neanderthal genomes from fossils and comparing them to modern human genomes, scientists found that people of non-African descent carry Neanderthal DNA, about 1 to 2% of their genomes. This interbreeding occurred after modern humans left Africa, probably in the Middle East, where the two groups encountered each other. Some Neanderthal genes provided benefits, like immune system variants, that helped fight diseases. These genes were favored by natural selection and remain common today. Denisovans, another ancient human group, also contributed DNA to modern populations. People in Southeast Asia, Oceania, and indigenous Australians carry Denisovan ancestry. Some Denisovan genes have been linked to high-altitude adaptation in Tibetans, showing that introgressed genes can provide specific advantages. These discoveries have rewritten human evolution. Rather than modern humans completely replacing archaic humans without mixing, there was interbreeding. We carry genetic echoes of these ancient encounters, evidence of a complex history of human interactions. Genes reveal the domestication of animals. Domestication transformed wild animals into the pets and livestock we know today. Genetic studies reveal when and where domestication occurred and what genes changed. By comparing genomes of domestic animals to their wild ancestors, scientists identified genes under selection during domestication. Dogs, descended from wolves, were the first domesticated animals. Genetic evidence suggests domestication began over 15,000 years ago. Genes affecting behavior, like those involved in fear, response, and aggression, show signs of selection. Physical changes, like floppy ears and coat color variations, are linked to genes that were also selected during domestication. Cats, pigs, cattle, chickens, and other domesticated animals each have their own genetic stories. Some were domesticated multiple times in different places. Others spread from a single domestication center. Genes for traits like milk production in cattle, docility in pigs, or egg-laying frequency in chickens show strong selection. Understanding domestication genetics informs modern breeding. It also reveals human history, as the animals we domesticated shaped our societies. The genetic changes that occurred during domestication are a testament to the power of selection, natural or artificial, to reshape genomes over time. Plants were also genetically transformed by domestication. Crops are dramatically different from their wild ancestors, the result of thousands of years of selection. Maize, for example, descended from a wild grass called teosinte. Teosinte seeds are tiny and hard, enclosed in a tough casing. Maize kernels are large, soft, and exposed on a cob. Genetic studies show that a handful of genes with major effects were selected during domestication, transforming teosinte into corn. Wheat, rice, tomatoes, and virtually all crops underwent similar transformations. Genes affecting seed size, fruit size, bitterness, toxin production, and ease of harvest, were targets of selection. Wild plants often have defenses like thorns, toxins, or bitter compounds that make them unpalatable. Domestication selected for reduced defenses and enhanced edibility. Some domesticated plants became so dependent on humans that they can't reproduce without help. Maize can't shed its seeds without human intervention. Bananas grown for consumption are seedless and must be propagated vegetatively. These plants are genetic partners of humanity, shaped by and dependent on us. Crop genetics informs modern agriculture. Understanding the genes that were selected during domestication helps breeders develop improved varieties. It also highlights genetic diversity in crop-wild relatives, which can be sources of useful traits like disease resistance or drought tolerance. Genes influence behavior in complex ways. Behavior has genetic components. Studies in animals and humans show that genes influence temperament, learning, aggression, and social behaviors. But behavior is also shaped by environment, experience, and culture, making it one of the most complex areas of genetics. Animal studies reveal specific genes affecting behavior. Mutations in certain genes can make mice more anxious or aggressive. Genes affecting neurotransmitters, hormones, and brain development all influence behavior. These genes don't determine behavior, but create tendencies and predispositions that interact with environment. In humans, behavioral genetics studies twins and families to estimate genetic influences. Twin studies compare identical and fraternal twins, estimating how much of the variation in a trait is due to genetics. Such studies suggest that traits like intelligence, personality, and even political attitudes have genetic components. But interpreting these findings requires caution. Heritability, the proportion of variation due to genetics, is specific to a population and environment. It doesn't mean a trait is fixed or unchangeable. And genes influence behavior through complex pathways involving brain development, hormone levels, and interactions with environment. Genes are part of the story, not the whole story. The genetics of intelligence is controversial. Intelligence, however defined, shows heritability in twin and family studies. This means genetic variation contributes to variation in intelligence. But what that means and how to interpret it is controversial. Hundreds of genetic variants have been associated with intelligence through genome-wide association studies. Each has a tiny effect. Together, they account for only a fraction of heritability, illustrating the complexity of the trait. Intelligence is influenced by many genes, each contributing a little, plus environmental factors like education, nutrition, and stimulation. Controversies arise when discussing group differences. Some have claimed genetic differences in intelligence between populations, but such claims are scientifically unsupported and ethically dangerous. Intelligence is a complex, multifaceted trait. Tests measure it imperfectly and are influenced by culture, language, and opportunity. Genetic differences between populations are minor, and environment plays a huge role. The genetics of intelligence matters for understanding neurodevelopment and cognitive disorders, not for making claims about groups. Every individual's intelligence is influenced by both their genes and their environment, and both are important for reaching one's potential. Genes and environment interact in development. Nature versus nurture is a false dichotomy. Genes and environment don't act independently. They interact constantly. Genes influence how an individual responds to environment. Environment affects how genes are expressed. The two are inseparable. A classic example is phenylketonuria. People with PKU have a genetic inability to metabolize phenylalanine. In a normal dietary environment, phenylalanine builds up and causes brain damage. But in a modified environment, with a low phenylalanine diet, they develop normally. The genetic trait only manifests in certain environments. Many traits show gene-environment interactions. Genetic susceptibility to certain diseases may only manifest under specific environmental conditions. A variant that increases skin cancer risk matters more in sunny climates. A genetic predisposition to obesity may have greater effect in environments with abundant high-calorie food and sedentary lifestyles. Understanding these interactions is crucial for medicine, public health, and education. Optimizing environments based on genetic knowledge could improve outcomes. Conversely, recognizing that environments shape genetic expression emphasizes the importance of providing supportive, enriching conditions for everyone. Epigenetics connects genes and environment. Epigenetics is the molecular bridge between genes and environment. Environmental factors, from diet to stress to toxins, can alter epigenetic marks, affecting gene expression without changing DNA sequence. These changes can influence health and development. Studies show that early life environment shapes epigenetic patterns. Maternal nutrition during pregnancy, stress, and exposure to chemicals can alter the developing fetus's epigenome. Some of these changes persist into adulthood, affecting disease risk or behavior. Animal studies provide striking examples. Rat pups that receive more maternal care develop different epigenetic marks and stress responses than those with less care. The difference is due to environment, but it's written in the epigenome and affects behavior throughout life. Whether such effects occur in humans to the same degree is an active area of research. But epigenetics underscores that genes are not fixed commands. They're responsive to the environment, adjustable, and dynamic. This responsiveness allows organisms to adapt to their conditions, fine-tuning gene expression for survival. The Human Genome Project mapped all human genes. Completed in 2003, the Human Genome Project was a monumental scientific achievement. It produced the first complete sequence of the human genome, identifying all 3 billion base pairs and mapping the location of genes. This reference genome became the foundation for modern genetics. The project was an international collaboration involving scientists from many countries. It took over a decade and cost billions of dollars. When completed, it provided a resource available to researchers worldwide, accelerating discoveries in medicine, biology, and evolution. One surprise was that humans have far fewer genes than expected. Early estimates were 100,000 or more. The actual count is around 20,000 to 25,000. We have only slightly more genes than simpler organisms like worms. Complexity arises not from gene number, but from regulation, alternative splicing, and interactions between genes. The Human Genome Project also revealed that much of the genome doesn't code for proteins. This non-coding DNA, once called junk, is now known to include regulatory elements, RNA genes, and structural sequences. Understanding this vast regulatory landscape is a current frontier of genomics. Genomic medicine is personalizing healthcare. Genomics is transforming medicine. By analyzing a patient's genome, doctors can tailor treatments, predict disease risks, and diagnose rare conditions. This personalized approach is already used in cancer treatment, pharmacogenomics, and rare disease diagnosis. In oncology, tumor genome sequencing identifies mutations that can be targeted with specific drugs. Patients receive treatments matched to their tumor's genetic profile, improving outcomes. As more targeted therapies are developed, genomic medicine in cancer is becoming standard. Rare disease diagnosis has been revolutionized by whole genome sequencing. Many rare diseases have genetic causes, but identifying the responsible gene was once nearly impossible. Now, sequencing a patient's genome can pinpoint mutations, providing diagnoses that end long diagnostic odysseys and guide treatment. Pharmacogenomics tailors drug selection and dosing based on genetic variants affecting drug metabolism. This reduces adverse reactions and improves efficacy. As pharmacogenomic testing becomes routine, prescribing will be increasingly personalized. The vision of genomic medicine is healthcare tailored to each individual's genetic makeup. It's not yet fully realized, but progress is rapid. As costs fall and knowledge grows, genomics will become a standard part of medical care. DNA sequencing has become fast and affordable. When the Human Genome Project began, sequencing DNA was slow and expensive. Completing a single human genome took years and cost billions. Today, a genome can be sequenced in hours for around $1,000. This dramatic progress has made genomics accessible. The key was new sequencing technologies. Next-generation sequencing machines can read millions of DNA fragments simultaneously, massively increasing throughput. Further advances, like nanopore sequencing, allow even faster and cheaper sequencing, with some devices small enough to hold in your hand. Affordable sequencing has enabled large-scale projects. Genomic databases now contain millions of genomes, providing resources for research. Direct-to-consumer testing has brought genetics to the public. Clinical sequencing is becoming routine for diagnosing diseases and guiding treatment. As sequencing becomes cheaper still, visions of universal genome sequencing emerge. Imagine every newborn having their genome sequenced, creating a lifelong health resource. Such efforts raise questions about data management, privacy, and equity, but they also promise unprecedented insights into health and disease. Bioinformatics analyzes vast genetic datasets. Modern genetics generates enormous amounts of data. A single genome sequence is billions of base pairs. Projects involving thousands of genomes produce petabytes of information. Analyzing this data requires bioinformatics, the application of computational tools to biological questions. Bioinformaticians develop algorithms to align sequences, identify variants, predict gene function, and find patterns in data. They build databases that organize genetic information and make it searchable. They create visualization tools that help researchers understand complex genomic landscapes. Machine learning and artificial intelligence are increasingly used in bioinformatics. These tools can identify subtle patterns that humans might miss, predict protein structures from sequences, or classify tumors based on genetic profiles. As datasets grow, computational power becomes as important as laboratory work. The synergy between biology and computation is transforming research. Questions that were once impossible to answer are now tractable. The bottleneck is often not generating data, but analyzing it, making bioinformatics a critical and growing field. Metagenomics studies communities of microorganisms. Your body hosts trillions of microorganisms, bacteria, viruses, fungi, and more. These microbes have their own genomes, and collectively they're called the microbiome. Metagenomics studies these communities by sequencing all the DNA in a sample without culturing individual species. The human microbiome influences health in profound ways. Gut bacteria help digest food, produce vitamins, and train the immune system. Imbalances in the microbiome are linked to diseases ranging from obesity to depression. Skin and mouth microbiomes affect susceptibility to infections and inflammation. Metagenomic studies reveal the diversity and function of these microbial communities. By sequencing DNA from gut samples, researchers identify which species are present and what genes they carry. This provides insights into how the microbiome contributes to health and disease. Beyond humans, metagenomics explores environmental microbiomes. Soil, ocean, and air all contain vast microbial diversity. These communities drive nutrient cycles, decompose organic matter, and influence climate. Understanding them through metagenomics reveals the hidden genetic diversity that sustains ecosystems. The microbiome is a second genome. Some scientists speak of the microbiome as a second genome. While your human genome is fixed at conception, your microbiome changes throughout life. It's influenced by diet, antibiotics, environment, and lifestyle. You can shape your microbiome, and it, in turn, shapes you. The genes in your microbiome outnumber your own genes by orders of magnitude. These microbial genes encode enzymes and pathways your cells lack, expanding your metabolic capabilities. Gut bacteria, for instance, break down complex carbohydrates that human enzymes can't digest, extracting nutrients from fiber. Research explores how to manipulate the microbiome for health. Probiotics, beneficial bacteria, are used to restore balance. Fecal microbiota transplants, transferring microbiomes from healthy donors to patients, have shown success in treating certain infections. Personalized dietary recommendations based on microbiome composition are being explored. The microbiome illustrates that we're not isolated individuals but ecosystems. Genetics must consider not just our own genes, but also the genes of our microbial partners. Health and disease arise from interactions between human and microbial genomes, a complex dance of coevolution. Horizontal gene transfer moves DNA between species. In most organisms, genes pass vertically from parent to offspring, but bacteria and some other organisms also transfer genes horizontally between individuals, even across species. A bacterium can take up DNA from its environment, receive it through viral infection, or directly transfer it to another cell. Horizontal gene transfer is a major force in bacterial evolution. It spreads antibiotic resistance genes rapidly through bacterial populations. A bacterium that evolves resistance can share the gene with others, even unrelated species. This is why antibiotic resistance spreads so quickly and poses such a challenge. Horizontal transfer also contributes to bacterial diversity and adaptation. Genes for metabolizing new substances, surviving extreme conditions, or producing toxins can spread through populations, allowing rapid adaptation to changing environments. In eukaryotes, horizontal gene transfer is rarer, but still occurs. Mitochondria and chloroplasts originated through horizontal transfer when ancient bacteria were engulfed and became symbiotic organelles. Some plants and animals have acquired genes from bacteria, viruses, or other organisms, though such events are uncommon and usually don't spread through populations. Antibiotic resistance is a genetic arms race. Antibiotic resistance is evolution in action. When bacteria are exposed to antibiotics, most die. But if a few have mutations or genes conferring resistance, they survive and reproduce. Their descendants inherit resistance, and the population becomes resistant to the antibiotic. Resistance genes spread through horizontal transfer, accelerating the process. A resistant bacterium can share its resistance gene with others, even across species barriers. This means that using antibiotics in one context, like agriculture or one infection, can promote resistance in bacteria elsewhere. The rise of multi-drug resistant bacteria is a global health crisis. Some infections are becoming untreatable with existing antibiotics. New drugs are developed, but bacteria evolve resistance to them too. It's an arms race, with human ingenuity pitted against bacterial evolution. Slowing resistance requires using antibiotics judiciously, completing prescribed courses, and preventing infections through hygiene and vaccination. Understanding the genetics of resistance helps identify mechanisms and develop strategies to combat it. But ultimately, evolution ensures that resistance will continue to emerge. Viruses have genomes too. Viruses are not cells, but they carry genetic information, either DNA or RNA. Viral genomes are tiny compared to cellular organisms, sometimes just a few genes. But those genes encode everything the virus needs to hijack a host cell and reproduce. When a virus infects a cell, it injects its genome. The viral genes commandeer the cell's machinery, directing it to produce viral proteins and copies of the viral genome. New virus particles assemble and burst out, infecting more cells. The process is efficient and, from the cell's perspective, catastrophic. Viral genomes mutate rapidly, especially RNA viruses. Without the proofreading mechanisms that cellular organisms have, errors accumulate quickly. This allows viruses to evolve fast, adapting to new hosts, evading immune systems, and developing drug resistance. It's why flu vaccines must be updated annually, and why HIV is so difficult to treat. Some viruses insert their genomes into the host's DNA. Retroviruses, like HIV, copy their RNA genome into DNA and integrate it into the host chromosome. This allows the virus to persist, hidden in the genome, reactivating later. Understanding viral genetics is crucial for developing treatments and vaccines. Viral sequences are embedded in human genomes. Remarkably, about 8% of the human genome consists of sequences from ancient retroviruses. These endogenous retroviruses, or ERVs, are remnants of infections that occurred in our ancestors millions of years ago. If a retrovirus infected a sperm or egg cell, its DNA could be inherited, becoming a permanent part of the genome. Most ERVs are now inactive, broken by mutations, but their presence is evidence of ancient infections. Some have been co-opted for useful functions. One ERV-derived gene is essential for placenta development in mammals, helping form the barrier between mother and fetus. Evolution repurposed viral genes for new roles. ERVs are shared across species in patterns that reflect evolutionary relationships. Humans and chimps share many ERVs, inherited from a common ancestor. These shared sequences are molecular fossils, recording the history of viral infections over millions of years. The fact that viral DNA is woven into our genomes shows how porous the boundaries between organisms are. Life is interconnected, with genetic material flowing between species, through infection, symbiosis, and, over deep time, incorporation into genomes. RNA can store genetic information. Most organisms use DNA as their genetic material, but some viruses use RNA. RNA, like DNA, can carry genetic information in its sequence of bases. It's usually single-stranded and less stable than DNA, but it's versatile, serving multiple roles. RNA viruses include influenza, HIV, coronaviruses, and many others. Their genomes are RNA molecules that encode viral proteins. When they infect cells, they replicate their RNA, sometimes converting it to DNA in retroviruses, or staying as RNA throughout their life cycle. RNA's role as genetic material is thought to be ancient. The RNA world hypothesis suggests that early life used RNA for both information storage and catalysis before DNA and proteins evolved. RNA can fold into complex shapes and catalyze reactions, properties that DNA lacks. Ribozymes, RNA molecules that act as enzymes, support this idea. Today, DNA is the dominant genetic material, but RNA remains essential. Messenger RNA carries information from DNA to ribosomes. Transfer RNA helps translate that information. Ribosomal RNA forms the catalytic core of ribosomes. RNA's versatility makes it central to genetics, even in organisms that primarily use DNA. Gene regulation is key to cellular identity. Cells in a multicellular organism all have the same genome, yet they look and function differently. This diversity arises from gene regulation, the control of which genes are active in each cell. Regulation occurs at many levels, from chromatin structure to transcription to protein stability. Transcription factors are proteins that bind to DNA and control whether genes are turned on or off. A cell type expresses specific transcription factors that activate genes appropriate for its function. Muscle cells express transcription factors that activate muscle genes. Neurons express factors that activate neuron genes. Enhancers and silencers, regulatory DNA sequences, modulate gene activity. They can be far from the genes they control, looping through space to interact with promoters. This three-dimensional genome organization is crucial for proper gene regulation. Errors in regulation cause disease. If a gene is expressed at the wrong time or place, development can go awry. If tumor suppressor genes are silenced or oncogenes are activated inappropriately, cancer can result. Understanding gene regulation is understanding how cells maintain their identities and respond to their environments. Transcription factors orchestrate development. During development, transcription factors act as master regulators, coordinating the expression of many genes. They turn on in cascades, with early factors activating later ones, creating waves of gene expression that pattern the developing embryo. The Hox genes, encoding transcription factors, are a famous example. They specify body segments, determining what forms where along the head-to-tail axis. Mutations in Hox genes can cause dramatic changes, like extra wings in flies or limbs in wrong places. Hox genes are conserved across animals, highlighting their fundamental importance. Other transcription factors specify cell types. MyoD, for instance, can convert fibroblasts, connective tissue cells, into muscle cells. Expressing a single transcription factor can reprogram a cell's identity, showing the power of these regulatory proteins. Understanding transcription factor networks helps explain how complex organisms develop from single cells. It also opens possibilities for regenerative medicine. If cells can be reprogrammed by manipulating transcription factors, damaged tissues might be regenerated, or new organs grown for transplantation. Alternative splicing increases protein diversity. A single gene can produce multiple proteins through alternative splicing. After a gene is transcribed into RNA, the RNA is processed. Non-coding regions, called introns, are removed, and coding regions, called exons, are joined together. But not all exons are always included. Cells can skip some exons, include optional ones, or use different combinations, producing different RNA molecules from the same gene. Each variant is translated into a different protein. This allows one gene to generate multiple proteins with related but distinct functions. Alternative splicing is widespread in humans. Some genes have dozens of possible splice variants. This increases the protein diversity of the genome, far beyond the 20,000 or so genes. It's one way organisms achieve complexity without vastly expanding gene number. Splicing is tightly regulated, and errors can cause disease. Mutations that disrupt splicing are a common cause of genetic disorders. Conversely, manipulating splicing is a potential therapeutic strategy. Drugs that alter splicing patterns are being developed to treat diseases caused by splicing defects. Non-coding RNAs regulate genes. Not all RNAs code for proteins. Many RNAs function as RNA molecules, playing regulatory roles. MicroRNAs are small RNAs that bind to messenger RNAs and prevent them from being translated into protein. They fine-tune gene expression, shutting down specific messages. Long non-coding RNAs, or LNC RNAs, are longer and more diverse. Some scaffold proteins together, others guide proteins to specific DNA locations, and still others regulate chromatin structure. Their functions are varied and still being discovered. These non-coding RNAs add layers of regulation to the genome. They allow cells to respond quickly to signals, adjusting protein levels without altering transcription. They're involved in development, differentiation, and disease. Dysregulation of microRNAs is implicated in cancer, heart disease, and neurological disorders. The discovery of extensive non-coding RNA has reshaped our understanding of the genome. Much of what was thought to be junk turns out to be transcribed into regulatory RNAs. The genome is not just a list of genes, but a complex regulatory network with RNA playing a central role. Chromatin structure affects gene accessibility. DNA doesn't float freely in the nucleus. It's wrapped around proteins called histones, forming nucleosomes. These nucleosomes coil into higher-order structures, creating chromatin. How tightly DNA is packed affects whether genes can be accessed and expressed. Tightly packed chromatin, called heterochromatin, is generally inactive. The DNA is inaccessible to transcription factors and RNA polymerase. Loosely packed chromatin, euchromatin, is accessible and can be actively transcribed. Cells regulate chromatin structure to control gene expression. Histone modifications, chemical changes to histone proteins, influence chromatin packing. Adding acetyl groups typically loosens chromatin, activating genes. Adding methyl groups can either loosen or tighten chromatin, depending on context. These modifications are part of the epigenetic code, marking which genes are active or silent. Chromatin structure is dynamic. It changes during development, in response to signals, and between cell types. Understanding chromatin regulation reveals how cells maintain stable identities while remaining responsive to change. It's also a target for therapies, as drugs that alter chromatin structure can reactivate silenced tumor suppressor genes or silence overactive oncogenes. Genetic mosaicism means not all cells are identical. Even within one individual, not all cells have identical genomes. Mutations occur during development and throughout life. A mutation in an early embryonic cell will be present in all descendants of that cell, creating a patch of genetically different tissue. This is mosaicism. Mosaicism can be harmless or consequential. If the mutation affects a gene important for development or growth, it might cause localized abnormalities. Some genetic diseases are mosaic, with symptoms depending on how many and which cells carry the mutation. Cancer is an extreme form of mosaicism. Tumor cells accumulate mutations, becoming genetically distinct from normal cells. Even within a tumor, different cells can have different mutations, creating genetic diversity that complicates treatment. Some mosaicism is normal and functional. Your immune system generates diversity by rearranging genes in immune cells, creating unique receptors in each cell. This genetic diversity allows your immune system to recognize countless pathogens. Mosaicism reminds us that genomes are not static. They change over time, creating genetic variation within individuals. This variation can be beneficial, neutral, or harmful, adding another layer of complexity to genetics. Chimeras contain cells from multiple individuals. A chimera is an organism containing cells from two or more genetically distinct individuals. Chimeras can arise naturally or be created artificially. Twin embryos sometimes fuse in the womb, creating a single individual with two genetic populations. People who receive organ transplants or blood transfusions are chimeras, carrying cells from donors. Mothers often carry fetal cells in their bodies decades after pregnancy, and fetuses carry some maternal cells. This microchimerism is common and usually harmless, though its effects are still being studied. Experimental chimeras are created in research. Cells from one species can be injected into embryos of another, creating animals with mixed tissues. Such experiments help study development and disease, and might one day enable growing human organs in animals for transplantation, though ethical questions abound. Chimerism challenges the notion of a single genome defining an individual. It shows that genetic boundaries are more fluid than we once thought. Like mosaicism, it adds complexity to our understanding of identity and genetics. Genetic testing of embryos allows selection. In vitro fertilization allows embryos to be genetically tested before implantation. Pre-implantation genetic testing analyzes cells from embryos, identifying those with chromosomal abnormalities or specific genetic mutations. Unaffected embryos can be selected for implantation, reducing the risk of genetic disease. For couples at risk of passing on serious genetic disorders, this technology offers the chance to have healthy children. It's used for conditions like cystic fibrosis, sickle cell disease, and Huntington's disease. It can also screen for chromosomal abnormalities, like Down syndrome, increasing the chances of successful pregnancy. The technology raises ethical questions. What conditions justify testing and selection? Where's the line between preventing disease and selecting for desired traits? Some worry about slippery slopes toward designer babies, where parents choose traits like intelligence or appearance. Regulation and ethical guidelines vary globally. Some countries allow broad use of pre-implantation testing, others restrict it. The technology itself is neutral, but its use reflects societal values. Conversations about its appropriate application are ongoing, balancing reproductive autonomy with ethical caution. Mitochondrial replacement prevents disease. Some genetic diseases are caused by mutations in mitochondrial DNA. Because mitochondria are inherited from the mother, women with mitochondrial mutations pass them to all their children. Mitochondrial replacement therapy aims to prevent this. The technique involves creating an embryo with DNA from 3 people. The nucleus from the mother's egg is transferred to a donor egg, whose nucleus has been removed, but whose healthy mitochondria remain. The egg is then fertilized with the father's sperm. The resulting child has nuclear DNA from both parents and mitochondrial DNA from the donor. This technology has successfully produced healthy babies in cases where mothers carried severe mitochondrial diseases. But it's controversial, sometimes called 3-parent babies, though the mitochondrial DNA contribution is tiny compared to nuclear DNA. Ethical concerns include the safety of the technique, the genetic contribution of the donor, and the implications of germline modification. Some countries have approved mitochondrial replacement under strict conditions. Others have banned it. It exemplifies the challenges of applying genetic technologies to human reproduction. Genetic diseases teach us about normal function. Studying genetic diseases reveals how normal genes work. When a gene is mutated and causes disease, The symptoms indicate what the normal gene does. Sickle cell disease showed that hemoglobin structure is critical for red blood cell function. Cystic fibrosis revealed a chloride channel essential for mucus consistency. Rare diseases, though individually uncommon, collectively teach us much about biology. Each is a natural experiment where one gene is altered. By studying the consequences, scientists learn what that gene normally does, how it interacts with other genes, and what pathways it's involved in. This knowledge benefits everyone, not just those with rare diseases. Understanding a pathway disrupted in one disease can reveal drug targets for more common conditions. Insights from rare genetic disorders have led to treatments used widely in medicine. Genetic diseases also drive technology development. The need to diagnose and treat these conditions has spurred advances in sequencing, gene therapy, and personalized medicine. Rare diseases are orphans in terms of patient numbers, but they're pioneers in genetic medicine. Gene therapy success stories are growing. Gene therapy was once a distant dream, but it's now reality for some diseases. Treatments have been approved for certain inherited blindnesses, spinal muscular atrophy, and blood disorders. More are in development, offering hope to patients with previously untreatable conditions. Luxturna treats a form of inherited blindness caused by mutations in the RPE65 gene. A working copy of the gene is delivered to retinal cells using a virus. Many patients have experienced dramatic improvements in vision, some seeing well enough to navigate independently for the first time. Zolgensma treats spinal muscular atrophy, a devastating disease where motor neurons die, leading to muscle weakness and death in infants. The treatment delivers a functional copy of the SMN1 gene. Infants treated early can develop normally, a transformation from what was once a fatal diagnosis. These successes come with challenges. Gene therapies are expensive, sometimes costing millions of dollars per patient. They're not cures for all. Some patients don't respond or experience side effects, but they represent proof of principle. Genetic diseases can be treated by fixing genes, a concept that seemed impossible a generation ago. CRISPR is being tested in human trials. CRISPR gene editing is moving from laboratory to clinic. Trials are underway for sickle cell disease, beta thalassemia, certain cancers, and inherited blindness. The goal is to precisely edit genes in patient cells, correcting mutations or disrupting disease-causing genes. In sickle cell and beta thalassemia trials, patients' blood stem cells are removed, edited to correct or compensate for the mutation, and infused back. Early results are promising, with some patients achieving normal hemoglobin levels and freedom from transfusions. Cancer trials use CRISPR to modify immune cells, enhancing their ability to recognize and attack tumors. Trials for inherited blindness deliver CRISPR directly to the eye, aiming to correct mutations in retinal cells. These trials are carefully monitored for safety. Off-target effects, where CRISPR cuts DNA in unintended places, are a concern. Long-term effects are unknown. But if successful, CRISPR-based therapies could treat or cure a wide range of genetic diseases, revolutionizing medicine. Regenerative medicine uses genetic reprogramming. Regenerative medicine aims to repair or replace damaged tissues and organs. Genetic reprogramming is a key tool. By manipulating gene expression, scientists can coax cells to change identity, differentiate, or even reverse differentiation. Induced pluripotent stem cells, created by reprogramming adult cells, offer potential for regenerative therapies. These cells can be differentiated into specific cell types needed for treatment. For instance, they could be turned into neurons to treat spinal cord injuries, or into heart cells to repair damaged hearts. Direct reprogramming converts one cell type to another without going through a stem cell stage. Fibroblasts have been converted into neurons, cardiomyocytes, and other cell types by expressing specific transcription factors. This approach could generate replacement cells for damaged tissues. Challenges include ensuring that reprogrammed cells function correctly and integrate safely. There's also risk of tumor formation if cells aren't properly controlled. But progress is rapid, and regenerative medicine, using genetic techniques, holds promise for treating injuries and degenerative diseases. Organoids, grown from stem cells, modeled tissues. Organoids are miniature, simplified versions of organs grown from stem cells in the lab. By providing the right signals, stem cells can be directed to self-organize into structures resembling brain, liver, kidney, or other tissues. These organoids mimic aspects of real organs, providing models for research and drug testing. Brain organoids, for instance, develop neurons and connections, allowing study of brain development and disorders. Researchers use them to model diseases like Alzheimer's or autism, testing potential treatments. They also study genetic mutations' effects on brain development, insights difficult to obtain otherwise. Organoids offer alternatives to animal models. They're human tissues, so they better reflect human biology. They also reduce reliance on animal testing, aligning with ethical goals. And they're personalized. Organoids can be grown from a patient's cells, carrying their genetic background, allowing tailored disease modeling and drug screening. Organoids aren't perfect replacements for whole organs. They lack blood vessels, immune cells, and connections to other systems. But they're valuable tools, bridging the gap between cell cultures and whole organisms, and advancing both basic research and therapeutic development. Ethics Guide: The Future of Genetic Technologies. As genetic technologies advance, ethical considerations become more pressing. Questions about privacy, consent, equity, and the limits of genetic manipulation require thoughtful answers. Society must grapple with what we can do versus what we should do. Genetic information is deeply personal. It reveals not just individual health risks, but also information about relatives. Balancing the benefits of genetic research and medicine with the need for privacy and informed consent is complex. Clear guidelines and strong protections are essential. Equity is another concern. Genetic technologies, especially expensive therapies, must be accessible to all, not just the wealthy. Addressing disparities in access ensures that the benefits of genetic medicine are shared broadly, reflecting societal values of fairness and justice. The power to edit human genomes, especially germline editing that affects future generations, raises profound questions. Who decides what changes are acceptable? How do we prevent misuse or unintended consequences? International cooperation and ongoing dialogue among scientists, ethicists, policymakers, and the public are necessary to navigate these challenges responsibly. Public engagement shapes genetic policy. Genetics affects everyone, not just scientists or patients. Public understanding and engagement shape how genetic technologies are developed, regulated, and used. Transparency, education, and inclusive dialogue ensure that policies reflect diverse values and concerns. Science communication is crucial. Helping people understand genetics, its possibilities and limitations, empowers informed decision-making. Misconceptions about genetics can lead to fear, stigma, or unrealistic expectations. Clear, accurate communication builds trust and enables constructive conversations. Public input into policy is essential. Decisions about genetic testing, data use, and technologies like CRISPR should involve those affected. Diverse voices, including ethicists, patient advocates, and communities enrich discussions and ensure that policies consider multiple perspectives. Genetics is a shared resource and responsibility. The choices made today will shape the future of medicine, agriculture, and society. Engaging broadly in these conversations ensures that genetics serves humanity's best interests, guided by wisdom, compassion, and respect for the complexity of life. The future of genetics holds immense promise. Genetics has come astonishingly far. From Mendel's peas to the Human Genome Project to CRISPR, each discovery builds on the last. The pace of progress is accelerating, with new tools and insights emerging constantly. The future promises even more transformative advances. We're on the cusp of routinely editing genomes to cure diseases. Personalized medicine, tailored to each individual's genetic makeup, is becoming reality. We can trace ancestry back thousands of years, understand evolution at the molecular level, and explore the genetic basis of complex traits. Challenges remain. Genetic diseases still await cures. Many questions about how genes and environment interact are unanswered. Ethical dilemmas require ongoing attention, but the trajectory is clear. Genetics will continue to deepen our understanding of life and expand our ability to improve health and well-being. The story of genetics is ultimately hopeful. It's a story of discovery, of learning how life works at its most fundamental level. It's a story of using that knowledge to heal, to feed the world, and to understand ourselves. As we continue reading this story, we write new chapters, guided by curiosity and responsibility. Genetics connects all living things. One of genetics' most beautiful revelations is that all life is connected. The genetic code is universal. Humans share genes with flies, yeast, and bacteria. We all descended from a common ancestor billions of years ago. This molecular kinship is profound. It means that studying any organism teaches us about life in general. A gene discovered in a worm might have a human counterpart with similar function. An insight from plant genetics might inform medicine. Life's diversity arises from variations on common themes, all written in the same genetic language. This connection also implies responsibility. The genes we carry are part of an unbroken chain stretching back to the origin of life and forward into the future. We're temporary custodians of genetic information accumulated over eons. How we use genetic technologies affects not just us, but future generations and the broader web of life. Genetics reveals unity in diversity. Despite our differences, at the genetic level, we're fundamentally similar. This shared heritage is a reminder of our common humanity and our place in the larger community of life on Earth. Every cell tells a story. Each cell in your body carries your entire genetic story. From the moment of conception, when two gametes merged their genomes, your genetic identity was established. Since then, through countless cell divisions, that genome has been copied and passed to every cell, carrying your history forward. Your genome is a mosaic of your parents' genomes, which were mosaics of their parents, stretching back through generations. It carries markers of ancient migrations, traces of interbreeding with archaic humans, and variants that arose through mutations over millennia. It's a record of your ancestry, written in molecules. But your genome is also your future. It influences your health, your responses to environment, and predispositions to traits and diseases. Understanding your genetic story helps you make informed choices about health, family planning, and life. And your genome is part of the ongoing story of life. The genes you carry will, if you have children, be mixed and passed on, contributing to the next generation's diversity. In this way, genetics connects past, present, and future in an unbroken thread. The genetic code is a language of life, Genetics is often described as a code, a language written in molecules. This metaphor is apt. DNA sequences spell out instructions, genes are like sentences, and genomes are like encyclopedias. Understanding this language has been one of humanity's great intellectual achievements. But genetics is more than metaphor. It's the actual mechanism by which information is stored, copied, and expressed in living things. It's chemistry and physics operating at the molecular level, creating the complexity and beauty of life. To read the genetic code is to read life's instruction manual. As we've learned to read this language, we've gained the ability to write it. We can edit genes, create synthetic DNA, and design organisms with new functions. This power brings responsibility. Like any language, genetics can be used for good or ill, to heal or harm. How we choose to use it defines us. The story of genetics is a story of humanity learning to read and write the language of life. It's a journey that began with curiosity and has led to profound knowledge and capability. As we continue this journey, may we do so with wisdom, humility, and reverence for the intricate code that animates every living thing. Closing reflections. Rest in the wonder of the code within. And so, dear listener, our journey through the world of genetics comes to a close. We've traveled from the double helix to CRISPR, from Mendel's garden to the depths of evolutionary time. We've explored how genes shape us, how they connect us to all life, and how they carry the story of our ancestors forward into the future. Genetics reveals that you are extraordinary. The genome within you is the product of billions of years of evolution, refined through countless generations. It's unique, a combination that has never existed before and will never exist again. Yet it also connects you to everyone and everything, written in a language shared across all of life. As you lie here now, perhaps your eyes are growing heavy, your breath slows into an easy rhythm, the day's thoughts begin to drift away, replaced by the quiet calm of approaching sleep. Allow yourself to rest in the wonder of it all. Imagine the trillions of cells in your body, each carrying your complete genetic blueprint. Picture DNA molecules coiled within every nucleus, silently holding the instructions that make you who you are. Visualize proteins folding, genes expressing, cells dividing in perfect synchrony, all orchestrated by the elegant code within. Feel a sense of connection to the vast tree of life. The genes you carry were once in your parents, your grandparents, ancestors stretching back through time. They passed through countless bodies, surviving ice ages and droughts, migrations and challenges, always preserved, always passed forward. You are part of this continuity, a link in the chain. And know that this genetic story continues, whether in your own cells as they repair and renew, or in future generations. If you choose to pass your genes forward, the story unfolds. Life persists, adapts, and evolves, written in molecules that have endured since the dawn of life itself. The genetic code is a reminder of connection, of complexity, and of the simple molecular elegance that underlies all existence. As you drift towards sleep, let these thoughts settle gently. You are part of something ancient and ongoing, something beautiful and mysterious. Rest in that knowledge. Rest in the quiet code within. If you happen to still be awake, you can go ahead and revisit any section that brought you peace, or simply let your mind wander where it will. But if your eyes are heavy now, if sleep is pulling you down into its gentle embrace, then go. Close your eyes, breathe deeply, and drift softly into stillness, into dreams, into the vast and timeless quiet, where the story of life continues, always in the elegant language of genes. Good night.